Hello future doctors, if you are looking for a one-stop, one-shot for the very first chapter of human physiology, that is breathing and exchange of gases, yes, you are at right place. So today in this video we are gonna complete every concept, every bits and bats, and myself, your zoology mentor Biswajit. After completion of this particular video, you can solve every question, every terminology and every concept from NCERT and from the chapter will be covered, right? And if you are thinking about for question practice, we will be with PPPs and also handwritten notes, yes. And for that you have to explore PW app, and from where you can get the link, just below, check the description box. So whenever we talk about the chapter breathing and exchange of gases, just one thing is cooking inside our mind, that how important is the chapter for NEET. Okay, so here I've kept a graph or a graph chart that you can visualize by yourself, that every year you can expect 2 to 3 important questions from this particular chapter. So it's about 8 to 12 marks, right? Okay, and what all the topics we are gonna cover today? So if you look your NCERT, here I've kept each and every topic starting from top to bottom, and we are gonna cover from basic to advanced. You know it very well. Okay, so starting from introduction, the anatomy and physiology of human respiratory system, respiratory organs, mechanism of breathing, a very conceptual thing to learn, respiratory volume and capacities. Again, many students, okay, most of the students, they find it very difficult to understand exchange of gases, transport of gases, the most difficult topic or most tricky topic from the chapter, and the regulation of respiration, and we'll be concluding it with 3 important disorders. Okay, so right here we are starting our introductory part of breathing and exchange of gases. So whenever we are putting our hand upon our chest, then we can feel that our chest is moving up and moving down during the time of inhalation and exhalation, right? So that is called movement related to breathing. Okay, now why we need to breathe every time? It's a biological rhythmic phenomena happening every time inside our body, right? So what is breathing basically? It's the process of exchange of oxygen from the atmosphere with carbon dioxide produced by the cells. So the process of exchanging atmospheric oxygen and cellular carbon dioxide, the whole and soul process, in normal language we will be calling it breathing, and there is a synonymous terminology is also given. You can also tell it at also commonly can express by the name of respiration, but there is a very important line of difference between breathing and respiration. We'll get to know. Okay, so you know that every day we are eating some food. Suppose today you have taken a burger, right? Now what are the components you will be finding in terms of food, in terms of nutrients? You will be finding protein, you will be finding carbohydrate, you will be finding fat, right? So inside the food, what we are getting? We are getting carbohydrate, we are getting proteins, we are getting fats, right? But there is a problem. After the ingestion of the food, if you directly approach these large molecules of carbohydrates, proteins and fats to your cells inside your body, you have nearby 100 trillions of cells present inside you. Now if you approach, please cell, oh my cell, please intake the food material whatever I have eaten. Now what will be the answer from the cell? Directly it will be rejecting it because it cannot absorb it and assimilate it inside the cytoplasm. So the next step your body need to take, that you have to break down. You have to break them down into simpler food material with the process of digestion. Now what digestion will be doing? With the help of some enzymes, it will be breaking down the food material, the carbohydrates, proteins and fats into glucose, into amino acids, and fats, if it is present in the form of triglyceride, it will be broken in the form of fatty acids. So now you can tell that this form of food, that is glucose, amino acid and fatty acids, they are simplified one. Now the cell will become happy suddenly and it will reabsorb the digested food materials in the form of glucose, amino acid and fatty acids, and it will be storing inside into the cytoplasm. Okay. Now what is the motto of eating? That is to derive energy from it. Now how energy will be produced? Suppose the cell has taken glucose. Now in order to you have to oxidize it. In order to harvest energy from the glucose molecule, you need oxygen from the atmosphere. So you need to give supply of atmospheric oxygen to the cell. Now oxygen will indirectly oxidize the glucose or break down in stepwise manner, and ultimately we will be getting carbon dioxide, water and energy, which will be produced in the form of ATP by the process of respiration. And as because the cell is breaking down a large molecule into a smaller one, like glucose is 6 carbon molecule and carbon dioxide is 1 carbon molecule, you can call the process as catabolism. All the chemical reactions taking place inside our cells, we call them metabolism. And metabolism can be of two types. Either you can make it or you can break it. If the cell is gaining dry weight, that is anabolism. If something destructive is happening or cell is losing its dry weight, here you can see glucose is broken into carbon dioxide. So that's why we are calling it catabolism. So ultimately the reactions, they are taking place inside the cell and producing harmful molecule like carbon dioxide. Now you need to expel out the carbon dioxide from the cell. Now the cell will be releasing the carbon dioxide. So this simple process of gaseous exchange, we are naming it breathing, and particularly the chemical reaction which is taking place inside every cell, we are calling it respiration. So here in the next slide, I have explained, I have given a table that you can easily differentiate between breathing and respiration, though sometimes we use as synonyms, right? So here you can observe that breathing is precisely a physical process, because while we are doing breathing, we have biological machine, and that is one pair of lungs inside our body, right? If you observe in fish, then they have gills inside their body. So they are exchanging oxygen and carbon dioxide with their body and water. We are exchanging oxygen and carbon dioxide with atmospheric gas and our body. So basically that's a physical process, and the respiration, as because we have seen, that is a catabolic chemical process. In breathing, merely there is no release of energy directly, but in case of respiration, energy will be released in the form of ATP, and that is the source of energy that every day, whatever work you are performing, whatever you are doing, I am teaching, you are listening, you are swimming, you are running. So from where the energy is coming? By the process, or the process which is happening inside your cell, and we are calling it respiration, and ATP, they are the energy currencies. Okay. For normal physical process, there will be no involvement of enzymes, but as because respiration is not a single line chemical reaction, it's a biosynthetic pathway. Multiple chemical reactions are taking place with the help of multiple enzymes. We call enzymes as biocatalysts. They help our body system to speed up any chemical reaction. Okay. Now the modes of breathing differ among different organisms, because different organisms are dwelling in different habitats. We are residing in terrestrial zone. Fish or aquatic animals, they are residing in water, right? So mode of breathing differs, but if we talk about respiration, particularly the organisms who are utilizing oxygen to break down the food material inside the cell, is called aerobic mode of respiration. So the process of aerobic respiration is same in all aerobic organism. If we talk about anaerobic respiration, that's a different topic. The difference between aerobic and anaerobic respiration you will be studying into the chapter respiration in organism. Okay. So let's talk about respiratory organs. Okay. Now whenever we have studied animal kingdom, it's highly diversified. Animal kingdom, there are near about 1.2 to 1.3 millions of species, right? And few of them are living in water, few of them are living in land, few of them are flying in air. So that is called, they have different habitats where they are living in. And we will be finding that in animal kingdom there are few animals who are quite primitive or ancient in nature, and there are few animals who are quite advanced. So that we call it the level of organization or the advancement of body design. So here it's saying that line of NCERT, the mechanism of breathing vary. Why it's varying among different groups of animals? So the question lies here. The mechanism of breathing, why it's varying among different groups of animals? So the answer lies here, depending mainly on their habitat, where they are living in. Habitat of a fish will be water, habitat of me and you will be land, or we are terrestrial animals. And the second thing, that is level of organization. What is level of organization? It's about the body design. Now if we talk about the body design of the level of organization, there are primitive animals where you will be finding their body is made up of multiple cells, but even tissue is absent, like in sponges. In some animals, those who are developed than sponges. If you compare like hydra, jellyfish, they have tissue inside them, but they don't have organs. There are some animals where you will be finding organs, but no organ system is present. And the highest order, the highest level of organization that we call organ system level, where body of animal is made up of multiple organ systems, right? So that is called level of organization. So habitat and level of organization will be deciding what will be the mode of respiration or mode of mechanism of breathing. So if we talk about sponges like porifera, coelenterata like hydra, jellyfish, and flatworms, they are platyhelminths. See, their body design is very much primitive or ancient. Their body is made up of a limited number of cells. So that's why they do not have any specialized respiratory structure, respiratory organ. So they use the process of simple diffusion and from where they are doing the gaseous exchange through their general body surface, through their general body surface. So primitive invertebrates, they are respiring through their body surface by the process of simple diffusion. Now if you ask me, sir, what is simple diffusion? See, if this is a chamber and you have taken a particular molecule, the concentration of the particular molecule is higher into this corner, and you are naming this corner as zone A, and rest the zone is zone B in this particular container. So what you will be finding, the tendency of these particles or molecules, they will be moving from the high concentration towards the low concentration zone, and that simple process is called simple diffusion. And the primitive organisms like sponges, they will be following this mode of simple diffusion. Where you will be finding sponges? Into aquatic environment, mostly they are marine. Their body has numerous opening or pores, that's why we are calling it porifera. So with the help of tiny pores, they will be intaking water, and with the help of large osculum, a large pore, they will be releasing the water. So whatever what actually they are doing, the sponges, with the help of tiny pores or ostia, they are intaking water, and with the help of large pore, they are releasing the water. So there will be continuous formation of water current inside and outside their body, and with the flow of water, what they are doing? They are doing exchange of oxygen and carbon dioxide. Similarly, we will be finding the same thing in hydra and jellyfish, and flatworms also. With the help of their body surface, they are exchanging carbon dioxide and water. Now let's talk about little bit developed animal. It is earthworm. They belong to the group annelids. Okay, so they are respiring through their moist cuticle. Now why they are respiring through their moist cuticle? See, if this is the body of earthworm, then that is basically protected or surrounded by a thin layer of cuticle that is moist in nature, and just beneath the skin we will be finding blood supply or blood vessel, highly vascularized skin. So if you are talking about respiratory surface, remember these two words, that is highly vascular. It has to be highly vascular, supply of blood vessel should be there, and it should be moist. So these two are the very important feature to qualify to become a respiratory surface, and the skin of earthworm having both the criteria. Beneath the skin supplied with blood vessel, and above the skin is covered by moist cuticle. So it will be easier for atmospheric gases like oxygen to get dissolved into that moisture and ultimately transport inside the blood, and the carbon dioxide which blood is carrying through the blood, it will be escaping through the skin and moist cuticle and will be released out into the atmosphere. Okay. Now we will talk about insects. Insects means cockroach, mosquitoes, butterfly. So they have network of tubules, and the tubules inside the body, they are responsible for delivering oxygen to each and every cells of their body, and those network of tubule we are calling it trachea, and these tracheas are also supported with tiny openings. Okay, they are called spiracles. And the respiration through trachea, we call it tracheal respiration. Okay, now I am explaining it, how they are doing respiration. You can take any example of any insect. Okay, so you will be finding they have tiny openings like our nostril into their abdomen and their thoracic region. These tiny openings we call them spiracles. So basically with the help of their spiracles, they are inhaling or they are exhaling, and if you go inside their body, you will be finding these spiracles are connected to branch network like tubes spread inside their body, and this branch network of tubes we are calling it trachea. Okay, now this obviously now we will be moving forward to higher order animals, those who are living in water. So now we will be talking about aquatic animals like aquatic arthropods. When you are talking about aquatic arthropods, shrimp, prawns, crabs, they will be coming into your mind. Aquatic mollusk are soft bodied animals. So most of the mollusks, they also dwell in freshwater or marine water like snails, right? And obviously fishes, they are exclusively aquatic. So all these aquatic animals, and along with you can also include tadpole larvae of amphibians, they respire through gills. Remember, aquatic animals respire through gills, and what about gills? They are special vascularized structure. Again, the gills are supported with rich supply of blood capillaries. Okay, and respiration through gills, we can call it branchial respiration, not bronchial. Respiration through gill, we will be calling it branchial respiration. Okay, now we are approaching towards higher order animals, those who are living in terrestrial zone or land. That is amphibians, there is frogs, reptiles means crocodiles, lizard, birds, crow, parrot, owl, mammals, human being, elephant, cow, buffalo, mice, rats. So amphibians, reptiles, birds and mammals, these land animals, they respire through one pair of lungs, and the mode of respiration we call it pulmonary respiration. From the Latin origin, pulmonary related to lungs, branchial related to gills, tracheal related to trachea. Okay. Now lungs, what are lungs basically? They are vascularized bags. The vascular word is very much important. Vascularized structure, gills. Vascularized bags, lungs. Network of tubes, trachea. You need to remember them in a very well manner. Okay. Now there is a special place for frogs. While their larval stage, like the larval stages of frogs, we call them tadpole, and they are exclusively aquatic in nature. You will be finding them into fresh water. So they have gills, but when they become adult after the metamorphosis, then you will be finding the frogs, they can respire through, obviously through their lungs, through their skin and through their buccal cavity. Remember it, it's very important to understand, very important to memorize. Frogs, the adult frogs, they respire through lungs while they are much active, they are jumping or running. They can respire through their skin while they are swimming inside the water, or even if they are performing their winter sleep and summer sleep. You won't be finding, like, frogs will be coming out from their burrows into monsoon, but during the time of peak summer and winter, they will be hiding inside their burrows, and we call that period, that inactive period, summer sleep or estivation, hibernation or winter sleep. And obviously in normal time, when they are inactive or they are just sitting quietly, they can respire through their mouth or buccal cavity or buccopharyngeal cavity. Okay, now let's talk about some questions. Questions are the report cards of our learnings or learning outcomes. Okay, so here is the first question. Find the pair which the given animal is incorrectly matched, okay, with their respiratory organs. Fish, they have gills, right? Now you just comment into the comment box that what we call the respiration mode of respiration through gills. Okay. Insect, so this is correct one. Insect, they have network of tubes. What do you call that network of tubes present inside the insect's body? Please comment down. And this is also correct match. Earthworm, they have moist cuticle, and remember one thing, moist cuticle means they're present upon skin, so you can also call it cutaneous. The animals who are respiring through their skin or moist cuticle, the mode can also be called as cutaneous respiration. Is not because they are very cute in nature, because their skin is covered by layer of moist cuticle. Okay. Now let's talk about mollusk. Do they have lungs? Wait a minute. No. Mollusks, I said they are mostly aquatic, so they have gills for respiration. So this is the wrong match and our correct answer. Okay. Another question, that is assertion and reason, is given here. So what's telling that insects have tracheal system for respiration? Yes, exactly. I was asking what you call the trachea, the network of tubes, right? So that is true for insects. Now reason is telling that insects have a network of tubes which helps them to transport atmospheric air within the body. Yes, directly that trachea. See, you will be finding that when you will be studying about cockroach, their blood is colorless. Why so? Our blood is red in color because our blood has hemoglobin, and that is acting as a oxygen carrying pigment, right? But most of the insects, you will be finding the blood is colorless, as because they don't have any respiratory pigment. because their respiratory network of tubes, trachea, directly transporting the oxygen to their tissue. So both assertion and reason are correct and correct explanation to each other. Now let's become doctor. Imagine after few months, after cracking the NEET, you will enter into some medical college, right? And in real life manner, you have to study human anatomy. So this is just a glimpse of that. We will be talking about human respiratory system. So let's get inside our respiratory system. So just imagine that you are not human, you are just a particle of air, and with that particle of air will be traveling inside the respiratory system of human being. Okay. So the air entering into our respiratory system with some holes here, and we call it external nostril. Okay. Now when we are getting inside the system, what we will be finding? The nose, the nasal cavity is divided into two tunnels, and we are calling it nasal passage. So inside the nose we have a nasal cavity, and a cartilaginous nasal septum is dividing our nasal cavity into two nasal passage. And what you are gonna get inside the nasal passage? You are gonna get some nasal hair, you are gonna get some mucosa cells to like who are secreting mucus. If you like put your hand inside your nose, you may feel it very creepy, but what you are gonna get? You are gonna get some mucousy coating. Is always wet inside the nose. So again, remember it, a respiratory surface always should be moist in nature. Okay. Basically, whatever air we are intaking, the air conditioning will like start taking place from your nasal cavity. And ultimately, the end of your nasal cavity will be there with the help of a nasal chamber. The roof of your nasal chamber, what will be getting here? There is a bone called nasal concha, and that is supported with a very special kind of neuroepithelium. We call it olfactory epithelium. While you are inhaling the air, the smell particles will be received here with the help of your olfactory epithelium. Remember it. Okay. Now here at the end of nasal chamber, the end part of your nasal chamber, what you are getting? You are getting your internal nostril. This is the end of your nose. See, when you will become a doctor and you will do specialization, then if you are doctor of ear, nose and throat, you will be calling it ENT specialist. Okay. So nose starts with external nostril, then you have your nasal cavity, you see, which is supported by mucosa epithelium, and also you will be finding olfactory epithelium for olfaction. So the word olfactory is related to smell. Okay. And ultimately it will be opened into nasal chamber, and the end part of your nose is through internal nostril. Now what after that? Now gradually with the air particles you are approaching down. Now here you found a common passage a common passage of air, a common passage of food and water, and that common passage we are naming it pharynx. So what is pharynx? Pharynx is common passage of air and food. So it's present into our throat region. Okay. And the pharynx, it has three parts. The part towards the internal nostril, we call it nasopharynx. The part of pharynx which is present towards the oral cavity, we call it oropharynx. The part of pharynx which is present towards the first part of your windpipe, we call or we name it sound box or larynx. It is called laryngopharynx. Okay. Now, so after pharynx, the air is gradually moving or proceeding further. Now the road has a bifurcation now. Toward the ventral surface. So this is the ventral side. This is the ventral side, and the back side is the dorsal side. Towards the ventral side, the pipe which is directly going inside the lungs, we call that windpipe, and the pipe which is going towards the stomach, we are calling it food pipe or esophagus. Okay. So what we will be calling the opening of food pipe or esophagus? We will be calling it gullet, and that is leading to food pipe or the esophagus. Now the initial part of your windpipe, we have named it larynx, or also you can call it sound or voice box. And how this larynx or voice box is made? That is made by cartilages. So it's a cartilaginous box which has some vocal cords to produce sound. That's why we also call larynx as sound or voice box. And the cartilaginous, as because we are calling it cartilaginous, how many cartilage it has? It has 9 cartilages. 3 are paired and 3 are unpaired. So total 6 plus 3, 9 cartilages are responsible for the formation of your larynx or sound box or voice box. Okay. Now let's expand the topic larynx. So that is the very initial part of your windpipe, cartilaginous in structure. Okay. 3 paired cartilage and 3 unpaired cartilages are there. Okay. Now why we are calling it sound box? As because it is made up of 2 false vocal cord. It has 4 vocal folds or vocal cords. Which cannot produce sound, and obviously it has 2 sound producing vocal cords. We call them true vocal cords. True vocal cords or vocal folds. So basically superior to vocal cords we will be calling it false, and inferior to vocal cords we will be calling them true vocal cords. So what these false vocal cords are doing, they are providing moisture and nourishment to the true vocal cords. So that's why sometimes if you are talking continuously, you just feel that if your throat is dry, you will feel that it's really hard to speak, right? So that time we need sip of water to make our these vocal folds moist. So moisture is very much important. Okay. So after larynx we have our windpipe, but at the entry point of larynx there is an opening, and we are calling that opening, we have named it as glottis. The entry point of larynx, the opening of larynx, we call it glottis. Now if you are like inhaling or exhaling, there is no problem, but the real problem arises when you are swallowing your food or water. Now during the time of swallowing, what will happen if a little portion of food enters into your respiratory tract? Then that will block the respiratory tract, that will collapse the respiratory tract, that will choke the respiratory tract, ultimately you may die. So Almighty has given a beautiful solution to that, that opening of our windpipe, that is glottis, is covered by a cartilaginous flap or cap, we call it epiglottis. Epi means upon, so it's a cap upon glottis, we call it epiglottis, which is made up of elastic cartilage. Okay, great. Now let's come down from the larynx. Now we will be finding a straight tube which is like going through your mid like ventral part of your thoracic cavity, and we are naming it our windpipe or trachea. And the trachea is supported by incomplete hyaline cartilage made C-rings or cartilaginous rings. So the trachea, which also can be called windpipe, that is supported by what? That is supported by, which is near about 10 to 12 centimeter long tube, and that is supported by what? That is supported by 16 to 20 cartilaginous ring. Now let's talk about trachea. So first of all, the theoretical thing we will be talking about trachea, that is supported by 16 to 20 incomplete cartilaginous rings or C-rings. Now, why this trachea is supported by the C-rings? The answer lies here: to prevent from getting it collapsed. See, this is trachea, and just behind that you have esophagus or food pipe, right? Now, while the food is moving inside the esophagus, then you will find a contraction and relaxation movement is happening that we call peristalsis. Peristalsis of esophagus. This is esophagus. Now that can create extra pressure on trachea, and that can make or that can form choking inside the trachea or windpipe, and ultimately if it get collapsed, then it will be really hard for us to inhale or exhale. So that's why it's supported by incomplete cartilaginous rings. So that is the scientific explanation. So if we observe it, like if we take a cut of this place and if you observe it, then what kind of view you can find here? Dorsally, it's supported by incomplete cartilage. At the ventral side, you have your esophagus. This is your trachea, this is your esophagus. Okay. Now let's move forward into our respiratory system. Now at a particular point, the trachea will get bifurcated and it will start forming bronchi, in singular bronchus. Now they will get inside the lungs. Okay. Now at what point? Nearby T5 or fifth thoracic vertebrae, the trachea get bifurcated to form bronchi. Now let's talk about the part where we are discussing about bronchi. See how it's going to happen. It's trachea near about fifth thoracic vertebra T5. It will get bifurcated and it will form two 1 degree or primary bronchi. Now that 1 degree or primary bronchi will get inside the lungs and we call them second degree or secondary bronchi. Now that secondary bronchi again will be forming branches and we will be calling that 3 degree or tertiary bronchi. First degree primary, 2 degree secondary, 3 degree tertiary. Okay. Now at the end of tertiary bronchi, you will find some narrow tubes, formation of some narrow tubes, we call them bronchioles. So first you will be finding initial bronchioles, and at the end of initial bronchioles you will be finding terminal bronchioles. And when these terminal bronchioles are also coming into the end, you will be finding respiratory bronchiole and ultimately some balloon-like structure, alveoli. Right? So the same thing I will be drawing it here. So if this is the trachea, at the place of T5 is getting bifurcated and forming two primary bronchi, and this primary bronchi, they will be entering into the lungs as left and right primary bronchus, and they will get bifurcated or trifurcated and we will be calling the branches of primary bronchi as secondary bronchi. Now if you observe the further branching, we call them tertiary bronchi. And suddenly the tertiary bronchi is getting narrowed down and we will be calling it initial bronchioles. And at the end of initial bronchioles, you will be finding terminal bronchiole. The beginning, the bronchioles, we will be calling them initial bronchioles, and at the end, you will be calling it terminal bronchioles. And even after the terminal bronchioles, you may find some thin respiratory bronchioles, and these respiratory bronchioles ultimately will be giving rise irregular balloon or sac-like structure which is surrounded by blood capillaries called alveoli. So that's all about the things which you will be finding inside your lungs in the form of a branched tree, right? Okay. Now we'll search for the cartilaginous rings. The trachea supported by cartilaginous ring, primary bronchi supported by cartilaginous ring, secondary bronchi supported by cartilaginous ring, tertiary bronchi supported by cartilaginous ring, initial bronchioles supported by cartilaginous rings. But you won't be finding cartilaginous ring into the terminal bronchioles, respiratory bronchioles, alveolar duct or alveoli. So cartilaginous rings can be found from trachea to initial bronchioles. Now if you are tracking the whole passage, whole pathway, then it's starting from external nostril. nasal cavity, nasal chamber, internal nostril, pharynx. After pharynx we have larynx. After larynx we have trachea. After trachea we have bronchi. After bronchi we have bronchioles. Starting from external nostril up to the bronchioles, we call this zone as conducting part of our respiratory system. Conducting part of our respiratory system. And what is the function of the conducting part of our respiratory system? Humidification of your air. Humidification of air, adjusting temperature of air to the body temperature, and clearing unwanted particles from the air. And this conducting part is not responsible for gaseous exchange. Remember it. What they are doing? Humidification of air, providing moisture to the air, adjusting temperature of the air with a body temperature, and obviously filtering or cleaning the air from the dust particles. Okay. So the conducting part is supported by a special kind of tissue called pseudo-stratified ciliated columnar glandular epithelium. Though the question won't be asked, it's extra information. The tissue will be finding inside the conducting part of our respiratory passage, pseudo-stratified ciliated columnar glandular epithelium. Okay. Few more things we need to understand here. See, starting from the trachea to initial bronchioles, you have the cartilaginous ring. Bronchi, when you are talking about bronchi, there are of three types: primary, secondary and tertiary bronchi. When we are talking about bronchioles, there are two types here: initial bronchioles and terminal bronchiole. And in the bracket I will be writing C if that part is supported with cartilaginous ring. Trachea, writing C, cartilaginous ring. 1 degree bronchiole have cartilaginous ring. 2 degree bronchi, I am extremely sorry, 1 degree bronchi, cartilaginous ring. 2nd degree bronchi have cartilaginous ring. 3rd degree tertiary bronchi have cartilaginous ring. Terminal bronchioles have cartilaginous ring. But in case of initial bronchioles, they have cartilaginous ring, but in case of terminal bronchioles, cartilaginous rings are absent. So here I can write it down in the form of words: from trachea to initial bronchioles, you will find cartilaginous rings, right? Okay, now what after terminal bronchioles? After terminal bronchioles, you have your respiratory bronchioles, though that is not mentioned in NCERT. So directly from terminal bronchioles, you may talk about alveolar duct and alveoli. What are alveoli? They are irregular sacs supported by blood capillaries. So this zone we are calling it exchange zone of your lung or respiratory zone of your lung. So what's happening here? If you observe a single alveolus, the singular form of alveoli, they are supported by blood vessel or blood capillaries especially. So with blood, exchange of oxygen and carbon dioxide is observed here. So that's why we are naming this zone as exchange zone or respiratory zone. So that was inside the lungs. So if we talk about the whole and soul lungs, a single lung, you will be finding bronchi, bronchioles, alveoli. So all together, bronchi, bronchioles and alveoli, the summation of all these things, we are naming it lung. And we have two lungs. We have two lungs. Larger right lung and smaller left lung. So we have two lung, right and left lung. Left lung is a bit smaller in compared to right lung. Why so? Because the apex of heart is present, or our heart is tilted towards the left. So that's why the apex of heart has occupied some place from this left lung. So it has to sacrifice some place, some volume. So that's why it's smaller in nature. One more important thing about the lungs, right lung is trilobed, left lung is bilobed. It has three parts or three lobes. Left lung has two parts or two lobes. So who is dividing lungs into lobes? Fisher. See, this is our right lung, this is our left lung. It has two lobes, it has three lobes. So the lobe formation will be done by fissure. This fissure is called oblique fissure. This fissure is also called oblique fissure. Fissure means a groove which is separating one part of lungs or one lobe from another lobe. And this fissure is called horizontal fissure. So, which is the common fissure? Oblique fissure. And here you can find a concave plane, we are naming it cardiac notch. In this cardiac notch, apex of heart is embedded. Got it? Okay. Now this is the famous diagram from your NCERT. Whatever we were discussing right now, everything is given in this diagram. But here we are going to talk, we know that our every lung is made up of 1 million very delicate thin balloon like structure called alveoli. So both the lungs are made up of 300 millions of alveoli, right? So it's very delicate structure. So during the time of breathing movement, inhaling or exhaling, there is a chance that friction may take place or force may generate, and that will cause shock towards your lung, and that may cause puncture of these alveoli. So that's why you need to protect your lung from any kind of friction or shock. That's why our lung is supported by, our lungs are supported by a double layered membrane called pleura or pleural membrane. Simply you can call it pleura or pleural membrane. As I said, that is double layered. So the covering of lung, we call it pleura. That is bilayered. It has two layers. See, the outer pleura or parietal pleura, the inner pleura or visceral pleura. So the pleura has two layers, outer pleura or parietal pleura, inner pleura or visceral pleura. Now what about this outer pleura? That is close to thoracic lining. This is close towards the thoracic cavity lining. And the inner pleura, that is close to lung surface. Two very important points, you have to mark it. Outer pleura is closer to thoracic cavity lining. The inner pleura, which is closer to your lung surface. Outer pleura closer to thoracic cavity lining, inner pleura which is closer to your lung surface. And what is present in between them? The outer pleura and inner pleura. So the space in between outer pleura and inner pleura is filled with pleural fluid. It's filled with pleural fluid, and that pleural fluid act as shock absorber or protect the lung surface from any kind of friction. So what is the function of pleural fluid here? Where I can write? Yes, I am writing it here. Pleural fluid protects lungs from friction or shock. So where these pleural fluids are present, if this is your lung, if this is the outer pleura, and if this is the inner pleura, so in between them the space, the pleural cavity, you will be finding that is filled with your pleural fluid. Okay. Now the very delicate and thin structure, alveoli, which are the structural and functional unit of lungs. Let's talk few words about them. So when you are talking about alveoli, they are approximately 300 million in number, we already know that. And in human lungs together forming a respiratory surface area of around 70 to 80 square meter. See, why so many alveoli? Why so many alveoli? Why not some few hundreds alveoli? The reason lies here, more the alveoli, more the surface area for efficient gaseous exchange. So more the alveoli, more the smaller alveoli, more alveoli, more the area for gaseous exchange. And do not forget that they are supplied with alveolar capillaries. Thousands of tiny capillaries, blood capillaries, will be surrounding the bunch of grape-like structure, or you may call them alveolus in singular and alveoli in plural. Okay. What about their internal structure? They are very thin, right? So I am making it little bit bigger. So first of all, you will be finding very flat cells, thin flat cells all over the alveoli. They are necessary for efficient gaseous exchange, and we are naming them pneumocyte type 1, and also few cells, oval or columnar or cuboidal shaped cells are present here. They are responsible for producing lecithin and protect the alveoli from choking. So these cells, we are calling it pneumocyte type 2. So alveoli, they have very thin surface. They have two types of cell, pneumocyte 1 and pneumocyte 2. Pneumocyte 1s are mainly responsible for this, providing surface for efficient gaseous exchange, and also this surface is supported by some WBC or phagocytic cells, and we call them dust cells. They will be clearing the debris here inside the alveoli. So that was all about the basic information regarding our structural and functional unit of lungs, that is alveoli in the place of gaseous exchange. Now let's solve some questions related to them. When there is no air in the initial bronchioles, you have learnt the initial bronchioles, they are guarded by some incomplete cartilaginous plates or rings, they do not collapse. Okay, if there is no air, they will not be collapsing. It's due to presence of lecithin? No. Lecithin, they will be produced inside the alveoli with the help of pneumocyte 2. Presence of pulmonary surfactant, this lecithin also acts as the pulmonary surfactant produced by again pneumocyte 2, so this is not our answer. Presence of incomplete cartilaginous rings, yes, as because they are supported by incomplete cartilaginous rings, there is no air even inside the respiratory passage, easily they won't be getting collapsed. Okay. Present of mucus, that is also not the answer. Obviously mucus will be present in throughout the respiratory surface, but that mucus helps the passage to become wet and help for the humidification of the air. So here we got the answer. Now let's move towards the next question. How many of the following structures are common in left and right lungs of humans? Okay, let's see. Horizontal fissure, you will be getting only right lung, so this is not a common structure. Oblique fissure, yes, you will be finding in both the lungs oblique fissure is present. Cardiac notch, only you will be finding in left lung, is absent in right lung, cardiac notch. Presence of lobes, yes, both the lungs are lobular. Obviously one lung is trilobed and another lung is bilobed, but lobes, the presence of lobes, this is the common feature for both the lungs. So how many of the following features are common? This one and this one. So option 2 is our answer. Okay. Now again a very important conceptual thing we are going to discuss now. Human respiratory system, and the topic is that we were talking about inside the lungs and outside the lungs. Okay. Now where these lungs are situated? So the lungs are situated inside the thoracic cavity. Okay. Now before diving into this topic, I'm giving you homework. Okay. And that homework you can comment in the below box. That homework is, sometime we are inhaling and sometime we are exhaling. Now it's related to sound production. We know that we have vocal folds or vocal cords which are helping us for sound production when there will be like vibration into them. But you can produce sound during the time of inhalation or you can produce sound during the time of exhalation. Just give me the answer into that comment box. Okay, just try it and let me know. Okay, now let's come again to our topic. So I was telling that so our lungs are floating, basically floating into our thoracic cavity. And again, here we will be finding what? A cage which is protecting the lungs and the organs situated here, and that we call thoracic cage. Now the question arises, the concept arises from this particular question, that can we directly alter the pulmonary volume? Is it like a balloon? Like we are sometime we have given so many examples that by showing a balloon, like our lung is working like balloon. But is it so? A balloon, the volume can be directly altered by blowing air or taking out the air from it. Like we have one pair of lungs and respiring through pulmonary mode of respiration. So like are they following the mechanism what balloon is following? That is, can we directly alter the pulmonary volume? So the answer will be a big no. We cannot directly alter the pulmonary volume. Why so? See, if you want to contract or relax something, then you must need the presence of muscle inside that. Now, when we are inhaling or exhaling, obviously, you need to volume up and volume down your lungs. But our lungs do not have muscles. Our lungs are devoid of muscles. No muscles are present into our lungs. So, directly we cannot alter the volume of lungs. So, the lungs, it need to rely upon the thoracic cage for volume up and volume down. So that's why it's written here a very important thing. Now we are going to talk about thoracic cavity. The thoracic cavity, that's a airtight chamber, and inside that airtight chamber your lung is present. Your lungs are not airtight, but inside the chamber, the cavity is there present, that chamber is airtight, and our lungs don't have its own muscles. Okay, that's why there is a direct connection between lungs and our thoracic cavity, and the very important formula I have written here: the volume of thoracic cavity is directly proportional to the volume of lungs. So if you want to alter the volume of lung, if you want to increase the volume of your lung, you have to increase the volume of your thoracic cavity. If you want to decrease the volume of your lung, you have to decrease the volume of your thoracic cavity. That's why I am telling the volume of thoracic cavity is directly proportional to the volume of lungs. The volume of thoracic cavity is directly proportional to the volume of lungs. Okay. See how our lungs work and how it's different from a balloon. If this is a balloon, so what you have to do when you are blowing, you have to push some volume of air inside that, and then you can feel that after some time the volume is increased. That's because you have inserted by force some volume of air inside that, and that follows the Avogadro's rule. Okay, that is about balloon. But what about our lungs? So if this is our lung, now you want to put some air inside that. So simply what you have to do, you have to create low pressure inside the lung. So from outside, which is in a high pressure zone, the air will enter. So for that, it's following Boyle's law, that is P is indirectly proportional to volume. So our lungs are following Boyle's law. So if you want to volume up or increase the volume of the lung, then what you have to do? First, you have to increase the volume. Once your lung volume is increased, the pressure is decreased. Now automatically the air will take entry into your lung. So who will be increasing the volume of your lung? Your thoracic cavity and the associated structures and muscle. So that's why our next topic is to study our thoracic cavity in a detailed manner. Again I am repeating, a balloon, the mode of mechanism of action of a balloon will be following Avogadro's law. The mode of mechanism of action of our lung will be following Boyle's law. First, you have to uh, insert volume inside the balloon, then you will see that gradually first you have to blow the air, then the volume will increase. But for our lungs, first you have to increase the volume. So when volume is increased, the pressure is decreased, so the atmospheric air will get entry inside your lung automatically. That's why also we call our lung sucking lung. Our lung basically sucking air. So that is the difference. Okay. Now, what are the structures of thoracic cavity? The structures will be finding of thoracic cavity. One flat bone, there is sternum. Numerous rib bones or ribs. Numerous vertebral bones. Most importantly, diaphragm. So together all this structure inside the thoracic cavity, it will be forming thoracic cage, and the lungs are encaged inside the thoracic cage. What is the positioning of the sternum? Here. So that is ventral to lungs. Their sternum is ventrally located. Rib bones laterally located sideways. Laterally located, lateral position. Vertebral columns dorsally located, and diaphragm. What is diaphragm? If this is our body, here we got our lungs. Just below the lungs there is a muscular membrane called diaphragm, which is separating our thoracic cavity from our abdominal cavity. So the location of diaphragm will be, this is the posterior part of our lungs. This is the anterior part of our body, and this is the posterior part of our body. So diaphragm, their location is posterior. Vertebral bones, their location is dorsal. Rib bones, their location is lateral. Sternum, their location is ventral. Now you can study this diagram drawn by me, obviously. So here you will be finding, this is the lung encaged inside the thoracic cavity. Laterally is surrounded by ribs. Ventrally it is surrounded by sternum. Posteriorly, this dome-shaped membranous diaphragm, and these structures which are present dorsally, the vertebral column. Okay. Now there are certain muscles which are associated with diaphragm. Certain muscles associated with diaphragm, we call them radial muscle. The muscles associated with diaphragm, we call them radial muscle. And the muscles which are associated or present in between the space of ribs, that we call intercostal space, we call that muscle intercostal muscle or ICM. The space between two ribs is called intercostal space, and the muscles present in between the ribs are called ICM, intercostal muscle. Intercostal Costal muscles, ICM, the muscles in between the ribs. So, why I am taking the names of these two important muscles, ICM and radial muscle? Radial muscle, they are associated with diaphragm, and ICM, they are associated with ribs. Basically, the movement of this muscle will be deciding the inhalation and exhalation movement of our chest or thoracic cavity. Okay. Now, at the end of this particular topic, we will be finding the steps involved in human respiration. Okay. Again, that's a concept. See, the air you are intaking, basically you are intaking oxygen. The air you are releasing, basically you are exhaling carbon dioxide. And this is the beginning step of our respiration, and the beginning step we are calling it pulmonary ventilation or breathing, and that involves exhalation or expiration, inhalation or inspiration. Okay. Now, the air with the help of your respiratory tract will get inside the lungs through trachea, primary, secondary, tertiary bronchi, and ultimately through initial and terminal bronchioles, it will be reaching inside the alveoli. You know very well, these alveoli are surrounded by numerous blood capillaries or alveolar capillaries. Now, this is the time for exchange of gases. Now, let's get inside the alveoli. Now, what's happening here? The oxygen from the air inside the alveoli will get inside the blood, and that will make the blood oxygenated. Because the oxygen is entering into the blood, and the deoxygenated blood which is coming with a lot of carbon dioxide is also released here inside the alveolar air. So, this particular process of gaseous exchange, the second part, that is diffusion. The first time the diffusion is taking in between the alveoli or alveolus and blood. Now, the blood, the oxygen will be carried through the blood and via heart that will be pumped towards the organs or systems of our body. Now, as soon as the oxygenated blood is approaching towards your organ, the blood will be releasing the oxygen, and the oxygen will diffuse inside the organs, and it will help in the process of cellular respiration. And as a result of cellular respiration, carbon dioxide are produced, and again the carbon dioxide will be loaded inside the blood. They will be diffusing from tissue or from organs towards the blood to make the blood deoxygenated. Okay. So, now in between this alveoli and tissue, what the blood is doing is acting as the mode of transportation. So, the third step will be transportation of gases. And the fourth step, that will be in between blood and tissue. In between blood and tissue, the gaseous exchange is there. So, again diffusion is taking place here. diffusion in between blood and tissue, and ultimately inside the tissue what you can observe, breakdown of the catabolic process, that is cellular respiration. So that's how, or that's why we can study this topic, and NCERT is telling, that's why NCERT is telling there are 5 steps of respiration. The very first step is breathing or pulmonary ventilation, which includes inspiration and expiration, or inhalation and exhalation. Okay. The second step, the diffusion of gases in between alveoli or alveolar membrane and blood, where oxygen is loaded into the blood and carbon dioxide is unloaded from the blood. Okay. Third step is transportation of the gases with the help of our blood and via heart, right? And the fourth step, another diffusion is taking place near to the tissue in between blood and tissue. In tissue, what is happening? Oxygen is unloaded and the blood is loaded with carbon dioxide. The opposite phenomena of the diffusion which has happened between alveoli and blood. And the last step of human respiration, the steps of respiration, the last step will be the utilization of that oxygen with just the tissues or the cells of the tissues got from the oxygenated blood, and utilization to break down the cellular glucose or amino acid, and that is called intracellular respiration. So let's solve some question with that like knowledge, whatever we have gathered now. So, okay, before solving the question, I need to tell you few more concepts. See, whatever happening in step 1 and step 2, there is pulmonary ventilation and diffusion between alveoli and blood. This step also can be called as external respiration. And the step 4, which is happening, the exchange of gases in between the blood and tissue or the cells, that can be called internal respiration. And obviously the chemical reaction taking place inside the cell, that is cellular respiration. So what is the difference between external respiration, internal respiration and cellular respiration? External respiration happening into alveolar level, internal respiration is the gaseous exchange into the tissue level, and the cellular respiration which is taking place inside the cell, which is actually we are calling it cellular respiration. Now with this knowledge you can solve this question. Assertion is telling that internal respiration involves gas exchange between the blood and body cells. Yes, it's absolutely true. Now the reason is telling cellular respiration involves conversion of food into energy. That is also true, but cellular respiration and internal respiration, they both are two different set of phenomena. So both assertion and reason, they are true, but it's not a correct explanation. Okay, please solve this question and answer me into the comment box. So question is, the trachea is a straight tube extending, yes, obviously remember it, 10 to 12 centimeter tube extending up to the mid thoracic cavity. The location of trachea is mid thoracic cavity, which divides that at the level of, yes, obviously we have prompt or we have discussed 2 to 3 times today's lecture, that they get bifurcated near to the fifth thoracic vertebrae into the right and left primary bronchi, and the location is also called, where they are getting bifurcated near to the T5, also can be called carina. Remember it. Okay, if you are saying, sir, we don't have any idea about vertebrae. So basically our back is supported, our trunk is supported by vertebral column, and this vertebral column is made up of 26 vertebral bones. So the first 7 are called cervical, so we call it C7, from C1 to C7, 7 cervical vertebrae. T12, 12 number of thoracic vertebrae. L5, 5 number of lumbar vertebrae, 1 sacrum or sacral vertebrae, and 1 coccyx or coccygeal vertebrae. So altogether we have 26 vertebral bones, and the trachea, like if you compare the vertebral location, that nearby fifth thoracic vertebrae, the trachea will be getting bifurcated. Now again solve this assertion and reason. Assertion is telling larynx is also known as sound box. Yes, obviously 100% correct. Larynx is sound box, voice box. Reason is saying it's a bony box. No, my dear, larynx is cartilaginous box. So assertion is true, but reason is false. Now let's solve this question again, one easier one. Select the sequence of steps in this respiration. So we know about the 5 sequential steps of respiration. So obviously first step will be pulmonary ventilation. Second step will be diffusion of gases across alveolar membrane. Third step will be diffusion of gases between blood and tissue. So it will be 4, 1, 2. Okay, the question is given the answer as A, B, C, D, E. Okay, so if it's A, B, C, D, E, D, A, B. Steps in respiration. Okay, no need to be getting worried. First it will be D, pulmonary ventilation, then DA, C, gaseous transportation, then B, diffusion of gases between blood and tissue, then lastly E, utilization of oxygen for catabolic process. So we got our answer, that is A. Now we are with a new question. Which is the site of actual diffusion of oxygen and carbon dioxide between blood and atmospheric air? Between atmospheric and air and blood, actual site of diffusion. Exchange part of respiratory system? Yes, obviously, the alveolar duct and alveoli zone. Conducting part? Never. Conducting parts from external nostril to terminal bronchiole, they don't take part in gaseous exchange. Respiratory, yes, synonymous term for exchange part of respiratory system. So we got our answer, that is for both A and C, both 1 and 3. Okay. So we are done with one important topic, and we are diving inside the most important and conceptual topic of the chapter, that is mechanism of breathing. So let's start. he is saying that breathing involves two stages. Yes, we know. You know, brother, there is inspiration and inhalation, and the another step is expiration or exhalation. Now, what happens during the time of inspiration? Atmospheric air is drawn in, air intaking atmospheric air, and during the time of expiration, alveolar air is released out. Simple thing, we know it very well. The movement of air into and out of the lungs is carried out by creating pressure gradient between the lungs and atmosphere. So that is the word I was searching for. So you have to create pressure gradient between lungs and atmosphere. So concept starts here. So few slides back I was telling one thing, that the whole and soul process is depending upon the pressure gradient. Air always moves from high pressure zone to low pressure zone. Air moves from high pressure zone to low pressure zone. Remember it. Okay. So what you have to do? You have to do a pressure gradient. You have to create pressure gradient between our lungs and atmosphere. Okay. Now, what is the atmospheric pressure? The atmospheric pressure is near about 760 millimeter mercury pressure. The atmospheric pressure is near about 760 millimeter mercury pressure, the air pressure. Okay. Now, if you are creating low pressure inside your lung, that we will be calling negative pressure. If you are creating high pressure inside your lung, in compared to atmospheric pressure, we will be calling it positive pressure. Now remember, our lung is negative pressure lung. Our lung is negative pressure lung, means if you want to inhale air, you have to create low pressure or negative pressure inside the lung, then only air will thrive inside your lungs. So for that, the formula is following. Already we have discussed, there is a derivation of Boyle's law. That is, pressure is indirectly proportional to volume. That means if you increase the volume, the pressure will be decreased. If you decrease the volume, the pressure will be increased. So that simple thing we have to create inside our lung with the help of the muscles we have spoken about during the time of discussing thoracic cavity or thoracic cage. Remember the muscles, intercostal muscles and the radial muscles of diaphragm. So this is the time for their utilization. Okay. So basic thing, you got it, is just about creating pressure gradient. If this part is having high air pressure and this part is having low air pressure, the air will move from high pressure zone to low pressure zone. So that is the nave of this concept. Okay. And while we will be utilizing our muscles to increase or decrease the volume of our thoracic cavity, the rib bones, most importantly, first to seventh pair of rib bones, or the rib bones which will be moving outwards during the time of inhalation, remember it, and upwards, and during the time of exhalation, the rib bones and the thoracic cavity will be moving inwards and downwards, just like the handle of this bucket. If you are inhaling, the rib will be moved up. If you are exhaling, the ribs will be moving down. Okay. And this is a detailed diagram of the thoracic cavity which we were talking about. So this is the very front bone, the flat bone, we are naming it the sternum. Associated, these are the rib bones. Right? Okay. From back side, these are the vertebral bones, that is dorsally placed, rib bones laterally placed, and if this is the location of lungs, just below it you have a dome shaped diaphragm. You have a dome shaped diaphragm. Okay. We need to track the movement of diaphragm during the time of inhalation and exhalation. And remember, I was telling about some muscles present in between the rib. The place in between the rib, we call it intercostal places. Intercostal space. And the muscles present into the intercostal space, we are naming them intercostal muscles. Okay. Now there are two types of intercostal muscles are there. There are two types of intercostal muscles are there. When you are talking about ICM, intercostal muscles, external intercostal muscle and internal intercostal muscle. So in normal breathing, we will be only talking about external intercostal muscle. When we discuss about forceful breathing, then we will be utilizing our internal intercostal muscle. Okay. Now let's again come into the topic. So it's time for exhalation. It's time for inhalation. Okay. So for inhalation, what we have to do? We have to create negative pressure, negative intrapulmonary pressure. Focus here, we have to create negative intrapulmonary pressure. Negative intrapulmonary pressure means low pressure inside the lungs in compared to air. So in air there will be high pressure. So what is the pressure of air? That is 760 millimeter mercury pressure. So if there is no breathing movement, the pressure inside our lungs is also similar or equal to atmospheric pressure, that is 760. Now you want to inhale. So for inhalation, what you have to do? You have to increase the volume of your lung. So as soon as the volume is increased, the pressure will be decreased. So if the volume is increased inside your lung, what will be the pressure? The pressure will be 758 mercury pressure. 758 mm, that is called, that also can be called as negative pressure. That also can be called as negative pressure. So how to create that? How to increase the volume of our lung? See, focus here. You have to contract. There will be contraction of EICM, external intercostal muscle. There will be contraction of the radial muscle of diaphragm. So when you are inhaling, there will be contraction in EICM and diaphragm, and ultimately the contraction of EICM will cause outwards or upwards movement of ribs and sternum. And if there is contraction in diaphragm, the diaphragm will become flattened. Normally the shape of diaphragm is like dome, and when there is contraction, like the radial muscle, they are creating force in the opposite direction, so the diaphragm will become flat shaped. So the flattening of diaphragm and the outwards movement, the outwards and upwards movement of the ribs and sternum. Here is written, the ribs and sternum are raised. The ribs and sternum are raised here. So that will increase the volume into the dorsoventral axis. So the contraction of EICM increases the volume into dorsoventral axis, and as soon as the diaphragm from dome shape it become flattened, so that will increase the volume into the anterior posterior axis. So diaphragm contraction increases the volume into anterior posterior axis. Again, listen it very carefully. The contraction of external intercostal muscle increases the thoracic volume into this axis, dorsoventral axis. The flattening of diaphragm or the contraction of diaphragm increases the thoracic volume into anterior posterior axis, and ultimately the volume is increased, and as soon as the volume is increased, we are following the Boyle's law, the pressure is decreased from 760 to 758. Now successfully we are able to create low pressure into the lung in compared to the high pressure of atmosphere, and the air will thrive inside from atmosphere inside the lungs. So that was all about inhalation. Now let's talk about exhalation. Now if you want to exhale, you have to create positive intrapulmonary pressure. Means the atmospheric pressure is remaining same, 760. The positive intrapulmonary pressure means from 760 you have to increase the pressure into 762. So that is called positive intrapulmonary pressure. So if you are successfully able to do so, the air will be exhaled out from the lungs towards the atmosphere. So here our motto is to decrease the volume to increase the pressure. Now how the volume will be decreased now? Whatever you did during the time of inhalation, whatever you did during the time of inhalation, during the time of exhalation you have to do just opposite. So here there will be relaxation of external intercostal muscle and relaxation of diaphragm. So what will be the outcome? If AICM is relaxed, the ribs and sternum will be moving backward and downward. And that will decrease the thoracic volume into dorsoventral axis, relaxed EICM. And what about relaxed diaphragm? When diaphragm is relaxed from a flattened position, it will go back to the dome shaped position. From a flat position, it will go back to the dome shaped position, and that will decrease the thoracic volume. That will decrease the thoracic volume into anterior-posterior axis. So ultimately, the successfully we are able to decrease the volume of thoracic cavity, and if the volume is decreased, the pressure automatically will be increased. So diaphragm will retain the shape or will become dome shaped. The diaphragm will become dome shaped. The diaphragm is relaxed and arched upwards. NCERT is telling that dome shape means this is the dome shape, and this is also can be tell that shape is arched, a bended arched shape or dome shaped. Got it? Moving forward, mechanism of breathing. Okay, so we have the ability to increase the strength of inspiration and expiration. Now the things we were telling, that was about normal breathing. So if you want to increase the strength, that is forceful breathing, you need to utilize some additional muscles. And what are those additional muscles? Those additional muscles are internal intercostal muscles and abdominal muscles. These muscles are utilized for forcible or forceful breathing. But in NCERT, we need to study only about normal breathing. Okay, and few more information is given here, that when you are breathing normally, the breathing rate is 12 to 16 times. The rate of breathing. Means in a minute, how many times we are inhaling and exhaling? 12 to 16 times. Let's solve few questions. Saying that the diaphragm plays a crucial role in the process of inhalation. Yes, obviously diaphragm is playing crucial role by changing its shape and like contributing into the volume. Okay, when the diaphragm contract, it flattens, okay, and increase the thoracic cavity volume. Yes, exactly right, leading to decrease in air pressure. Yes, absolutely correct reason. So both assertion and reason, they are correct, and the assertion, the reason is correct explanation for the assertion. Okay, now again another conceptual topic we are entering, that is respiratory volume and capacities. Respiratory volume and capacities. Okay, so the volume of air involved in breathing movement can be estimated by using a machine called spirometer, which helps us for clinical assessment of pulmonary functions. We are coming into that. Let's talk about the respiratory volumes. So we need to talk about 4 respiratory volumes. We need to talk about 4 respiratory volumes. First is tidal volume or TV. The second volume is expiratory reserve volume, or ERV. Third one is inspiratory reserve volume, or IRV, and the fourth one is residual volume, or RV. Okay. Now what about tidal volume? This is the normal, during the time of normal breathing, the amount of, or the volume of air we are inhaling or exhaling. So it's near about 500 ml per inhalation and per exhalation. This is the volume of air we are inhaling or exhaling in normal breathing. (500 ml). Now if someone asks you what will be the tidal volume in a minute, so in a minute we can inspire and expire 12 to 16 times. So you can conclude in a minute the volume will be 6000 to 8000 ml per minute. So tidal volume per breathing, per inhalation, per exhalation, or tidal volume per minute. You just whenever you are getting the question, you just read the question carefully. Okay. So now expiratory reserve volume, ERV, multiply 2 with tidal volume, you will be getting 1000 ml. So that is your expiratory reserve volume. And how you can define it? The volume of air can be expired out after a normal, releasing a normal tidal volume upon a forcible exhalation. So the volume of air can be exhaled on forcible exhalation. And you can add this line over a tidal volume. Means you are exhaling, you have exhaled normal 500 ml of air, that is tidal volume. Now over that, after that, if you are doing forcible exhalation, you have become Ramdev Baba. So the extra 1000 ml of air you can expire out. So that is your expiratory reserve volume. Now let's come to the inspiratory reserve volume. Multiply 3 with the expiratory reserve volume. That is, we are getting 3000 ml. That is ERV, that is IRV, or inspiratory reserve volume. Now that is the amount of the volume of air we can inspire on forcible inspiration over a tidal volume. Like you have inspired tidal volume 500 ml. Now over that, if you want to inhale more, that more 3000 ml air you can inhale. Now ultimately the last one, that is residual volume, that is 1200 ml residual volume. If you want to like if you are even doing the most forcible expiration, you have become Ramdev Baba. Even Ramdev Baba cannot exhale out this minimum volume of air from your lungs. They always reside inside your lungs. So that's why we are calling it residual volume. The volume of air always remain inside lungs even after most forceful expiration. Okay. Now to understand it easily, I am giving you one example. Suppose there is a classroom, and in that classroom there are total 57 seats for students, right? Okay. Total like enrollment of 57 students are there. There are 12 students, they attend each and every class. They never miss the class for anything or everything. Even it's storming outside, raining outside, they attend each and every class. So these 12 students you can compare with the residual volume. Okay. There are 10 students inside the class. They also really attentive. They really like from inside they attend each and every class. But sometime they won't be like coming with their homework. So the teacher forcefully expelling them out. Sometime it happens. So these 10 students you can compare them with the expiratory reserve volume. Okay. Now there are 5 students, they will come one day and another day they will be absent. They will come one day, another day they will be absent. Now this 5 students you can compare with the 500 ml air, that is tidal volume, that is coming going out, coming in, going out, inhaling and exhaling. And there are 30 students, they really don't have any intention to attend any of the classes, but sometimes their parents forcefully telling, please go and attend the classes. So when you are forcing them, they will come and attend the class. So these 30 students, compare this 3000 ml air, that is inspiratory reserve volume. You have to forcefully inhale them. Now let's come to the respiratory capacities, which is related to, which is a derivative of the respiratory volume. But before diving into the respiratory capacities, I'll be talking about one more volume, that is anatomical dead space volume. That is not related to the lung volumes. It is totally different thing. What is anatomical dead space volume? There will be always 150 ml volume of air present into the conducting zone of our respiratory system. So conducting zone, that is not related to the gaseous exchange. So this volume is also not related to your respiratory volume. So 150 ml volume of air is called anatomical dead space volume. This is the volume of air always we are finding inside the conducting air passage inside our respiratory zone. Remember it. Okay. Now let's come to the respiratory capacities. There are 5 capacities we need to understand in a very well manner. Okay. So first is inspiratory capacity. So what is inspiratory capacity? See, the capacities we need to calculate them from the data or from the formulas which we have studied into the respiratory volumes. There are 4 respiratory volumes, remember them. So this is the total volume of air a person can inspire after a normal expiration. So what is the total volume of air a person can inspire? A person can inspire tidal volume, a person by forcefully can inspire inspiratory reserve volume. So if you add these two, you will be getting your inspiratory capacity. Now what is expiratory capacity? The total volume of air a person can expire after a normal inspiration. So after a normal inspiration, what volume of air a person can inspire? A person can expire tidal volume 500 ml plus expiratory reserve volume 1000 ml. So TV plus ERV you will be getting expiratory capacity. Now functional residual capacity. What is functional residual capacity? The volume of air that will remain in the lungs after normal expiration. So if you are doing normal expiration, only you have exhaled out 500 ml. You are not forcibly expiring, you are forcibly exhaling. So you have the residual volume inside your lungs, you have the expiratory reserve volume inside your lungs. So when you add RV plus ERV, you will be getting your functional residual capacity. Now let's come to the vital capacity. See, remember the word, remember the sentence. Virat wears tie. Virat Kohli, you know very well. Virat is wearing tie. TIE. Tidal volume plus inspiratory reserve volume. Tidal volume plus inspiratory reserve volume plus I. TIE. Expiratory reserve volume. So from the 4 volumes, if you exclude the residual volume, by adding TV, IRV, ERV, you will be getting the vital capacity. Okay. Now how to explain vital capacity? So the explanation is here. The maximum volume of air a person can breathe in after a forced expiration. So after forced expiration, you have only residual volume left inside your lung. Now if you are doing a most forceful inspiration, So you can inhale the ERB, you can inhale the TV, you can inhale the IRV. So that is the total volume of your vital capacity, or vice versa. After most forceful inhalation, your lung is full. Now if you are doing most forceful exhalation, first you will be exhaling your IRV, then you will be exhaling your tidal volume TV, then you will be exhaling your ERV. Then only residual volume is left. So the amount of the volume of air you are releasing, you are exhaling, that is your vital capacity. So it can be explained in both the way. After most forceful exhalation, if you are performing most forceful inhalation, or vice versa. Okay, you have to calculate, you have to add TV plus IRV plus ERV to get the vital capacity. Now last it comes with the total lung capacity, or TLC. So you just have to do what? You just have to add TV plus IRV plus ERV plus RV. So all the 4 volumes you have to add to get your total lung capacity when your lung is full, right? Or else there is another formula. We know that TV plus IRV plus ERV, they are vital capacity. So VC plus RV, vital capacity plus residual volume, is also can be the formula to get total lung capacity. Okay, now let's solve some question related to the topics. Which of the following best describe the functional residual capacity? So what was functional residual capacity? After normal expiration, the volume of air will be getting inside our lungs. Okay, so the answer we will have to find here. The total volume of air a person can inhale after normal expiration. After a normal expiration, the total volume of air a person can inhale, that is not functional residual volume, that is inspiratory capacity. The volume of air that remains in the lungs after normal expiration, the volume of air that remains in the lungs, that is after normal respiration. So you have your residual volume and expiratory reserve volume. So this is the answer. Option 3 is saying the maximum volume of air a person can exhale after forced inspiration. So here they are talking about vital capacity. Option 4, the maximum volume of air a person can breathe in after a forced expiration. The maximum volume of air a person can breathe in after a forced, again that is they're telling about the vital capacity. Now come to the next question. Which of the following volume is not included in the vital capacity? Vital capacity is excluding the stagnant one, the remaining one, that is residual volume. And again a very important thing, that residual volume cannot be measured with spirometer. Okay, so is there any machine which can measure the residual volume? Yes, there are multiple other techniques which is not given in NCERT obviously, but with normal spirometry we cannot measure the residual volume, but we can measure the tidal volume, inspiratory reserve volume or expiratory reserve volume and all the capacities. Okay, now we will moving forward to a new topic, that is gaseous exchange and diffusion of respiratory gases. So gaseous exchange occur in 2 levels, first at alveolar level and second at tissue level respectively, which depends on difference of partial pressure primarily across the diffusing surface. Now here, when a gas is traveling from one medium to another medium, that who are the guiding factors or affecting factors or the important factors that are involved into the rate of their diffusion. When the gases are diffusing or traveling from one medium to another medium, the factors which are guiding them, that from where to where they have to diffuse in or diffuse out. So there are some factors. First factor is solubility of gases. Second factor is partial pressure of gases. Third factor, thickness of diffusion membrane. And the fourth factor is the area of diffusion surface. So we need to discuss each and every factor. First factor is solubility of gases. So there is a very limited thing to discuss inside the solubility of gases. So if a particular gas is more soluble or having higher solubility, that will diffuse or that will get dissolved into our blood more rapidly. That is the funda. So here we can see that carbon dioxide is having, CO2 has 20 to 25 times higher solubility than oxygen. So just for the sake of information and concept, you need to remember it. The solubility difference between carbon dioxide and oxygen. Carbon dioxide is having higher solubility, and how much higher, how much more? 20 to 25 times more soluble in water in compared to oxygen. Now, why we are talking about this, the solubility or comparing between the solubility? See, ultimately inside our body, though we are inhaling air and that contains all the gases, but the relevant gases are carbon dioxide and oxygen. Nitrogen is quite irrelevant for our biological system, right, in terms of exchange. Okay, so the carbon dioxide will dissolve more rapidly into the water which is present into the plasma of our blood. Ultimately, the solubility is how fast they can get dissolved into the water. And what is the importance of water? We know that blood is made up of 55% of plasma, and that plasma is made up of again 90 to 92% of water. So ultimately the gases when they are going inside our blood, their first choice to get dissolved into the blood of the plasma. Okay. Now let's talk about the partial pressure of gases. Now what is partial pressure? So if this is a closed chamber and air is present, air particles are present inside that closed chamber. So the normal pressure of air, what do you know the normal pressure of air? The air pressure is 760 millimeter mercury pressure. Okay. And that air has many gases. Air has nitrogen, air has carbon dioxide, air has oxygen, air has carbon dioxide and other gases. The percentage wise, nitrogen is 78%, oxygen 21%, carbon dioxide 0.03%. So there is the percentage wise volume of different gases into the air, and air is a mixture of different gases. Okay. Now suppose these are the nitrogen particles the most abundant gas into the air. This green, they are the oxygen particles, the second most abundant one, and this purple, this is the carbon dioxide, very less. And these gaseous particles, they are showing Brownian movement. Randomly they are moving and they are colliding with the surface of that container and creating a pressure, that is called particular pressure of the individual gas. So all together all these particles are exerting pressure on the container surface, and every individual gas also exerting its own pressure. So individual pressure of a particular gas, we are calling it as partial pressure. Now how to calculate partial pressure of gas? If you want to calculate partial pressure of nitrogen, so will be exhibiting as Pn2. So you have to calculate 78% of 760 and you will be getting somehow nearby 590. If you want to calculate partial pressure of oxygen, 21% of 760, somehow you will be getting 159. If you want to calculate partial pressure of carbon dioxide in air, you have to calculate 0.03% of 760 and you will be getting 0.3 mmHg. So that's how partial pressure is calculated. Okay. Now we also have to look the partial pressure of these two gases, particularly oxygen and carbon dioxide, inside our body. Okay. So if we talk about the atmospheric air, the partial pressure of oxygen is 159 mm mercury pressure and partial pressure of carbon dioxide is 0.3 mm mercury pressure. Now from alveoli, every part is present inside the body. Now remember it, I am telling you one trick. Inside our body, either it's oxygen or carbon dioxide, if it's weaker than other, the partial pressure you keep it 40. So in alveoli, who is stronger? Oxygen is stronger, carbon dioxide is weaker. So partial pressure will be 40. In deoxygenated blood, who is stronger? Oxygen is weaker and carbon dioxide is stronger. So weaker one is having partial pressure 40. In oxygenated blood, who is weaker? Carbon dioxide is weaker. The partial pressure will be 40. In tissue, who is stronger? Respiration is going on. So carbon dioxide is stronger. So weaker oxygen is having partial pressure in tissue is 40. Now what about carbon dioxide? Wherever carbon dioxide is stronger than oxygen inside our body, the partial pressure you keep it 45. So in tissue, carbon dioxide is stronger, partial pressure is 45. Deoxygenated blood, carbon dioxide is stronger, partial pressure is 45. Now you have to put the value of oxygen, partial pressure of oxygen in alveoli and oxygenated blood. In alveoli, the partial pressure of oxygen is 104 mm mercury pressure, and in oxygenated blood, the partial pressure of oxygen is 95 mm mercury pressure. So we got a proper table. Now let's justify this table. How this is important for gaseous exchange? See, in atmosphere, 159 is quite high pressure in compared to the alveolar air, alveolar oxygen. So oxygen will move from atmosphere to alveoli. Now in alveoli who will come? Deoxygenated blood will come, which is having partial pressure of oxygen that is quite low, 40. So alveoli now, if you compare partial pressure of oxygen in alveoli and deoxygenated blood, this is high pressure, this is low pressure. So again oxygen will enter from alveoli to deoxygenated blood and that will make the deoxygenated blood oxygenated. So the partial pressure of oxygen is increased from 40 to 95. Okay. Now that oxygenated blood will travel towards the tissue, and inside the tissue the cells are performing cellular respiration. So the partial pressure of oxygen is very less there. So oxygen again will thrive from oxygenated blood, that is high pressure, to low pressure tissue. Now what will be the fate of carbon dioxide? So here is to follow this passage, carbon dioxide is having high pressure in tissue, and the blood which is approaching towards the tissue, that is oxygenated blood, that is having a low pressure of carbon dioxide. So the carbon dioxide will travel from high pressure to low pressure zone, and that will make the oxygenated blood deoxygenated. From 40 to 45 again the partial pressure of carbon dioxide has increased here. Now the deoxygenated blood will move towards the alveoli. Now inside the alveolar air the partial pressure of carbon dioxide is very less, that is 40, and the deoxygenated blood the partial pressure of carbon dioxide is higher comparatively. So carbon dioxide will move from deoxygenated blood towards the alveoli, and again from alveolar air to atmosphere, because again it is getting a passage from high pressure zone to low pressure zone. That's why we are telling that we are inhaling oxygen and we are exhaling carbon dioxide, right? Okay. Now a particular diagram related to this is given. Okay. So when we are talking about the air or atmosphere, the partial pressure of oxygen is 159 and carbon dioxide is 0.3. So that's why there is a proper pressure gradient is formed here. So oxygen will go inside, as because in the alveolar air the oxygen partial pressure will be 104, that is less than the air. So this particular table is given in a diagrammatic format. And inside the alveoli the partial pressure of carbon dioxide is 40, and in atmosphere the partial pressure of carbon dioxide is 0.3. So what will be the passage of carbon dioxide? The carbon dioxide will go out. Okay. Now towards this alveoli the deoxygenated blood is approaching with the help of pulmonary arteries. Okay. Now what is the partial pressure inside that deoxygenated blood? Oxygen is having partial pressure 40 and carbon dioxide is having partial pressure 45. So what it will observe, inside the alveolar air the oxygen is 104. So oxygen will move from high pressure zone to low pressure zone, and carbon dioxide will move from high pressure zone to low pressure zone, but the sequence is in opposite side. Every time the air or the blood is calculating the partial pressure of oxygen and carbon dioxide, and that will decide the direction of the flow. And by gaining oxygen the blood will become oxygenated, and the oxygenated blood will be transported by pulmonary veins towards the heart, to the left atrium of the heart. Now the oxygenated blood will reach from left atrium to left ventricle. The left ventricle will pump the blood with the help of systemic arteries, which is carrying oxygenated blood with partial pressure of oxygen 95 and partial pressure of carbon dioxide 40. And this oxygenated blood will be approaching towards the tissue, and inside the tissue there is high pressure of carbon dioxide. There is inside the tissue the partial pressure of carbon dioxide is very high. The partial pressure of carbon dioxide is 45 inside the tissue, and partial pressure of oxygen is only 40 inside the tissue. So the oxygenated blood will lose oxygen here, and the oxygenated blood will gain carbon dioxide, and that will make the venous blood deoxygenated. So that's how while the blood circulation is happening inside our body, okay, gaseous exchange taking place into the alveolar zone and tissue zone depending upon their partial pressure gradient. And remember one thing here, every 100 ml oxygenated blood, every 100 ml oxygenated blood carrying 20 ml of oxygen and 48 ml of carbon dioxide. Every 100 ml of deoxygenated blood carrying 15 ml of oxygen and 52 ml of carbon dioxide. Again I am telling, every 100 ml oxygenated blood have 20 ml oxygen. Every 100 ml deoxygenated blood is having 15 ml of oxygen. So how much oxygen is delivered to the tissue? 5 ml oxygen is delivered to the tissue from 100 ml blood. Now let's compare to the carbon dioxide volume. Every 100 ml oxygenated blood has 48 ml carbon dioxide. Every 100 ml deoxygenated blood has 52 ml of carbon dioxide. Now while the carbon dioxide is getting loaded from tissue to blood and making it deoxygenated, how much carbon dioxide is entering into the 100 ml blood? 4 ml carbon dioxide is loaded into the blood, and this carbon dioxide will be unloaded here into the alveoli. So 5 ml oxygen is entering into the tissue level from blood to tissue, and 4 ml carbon dioxide is loading inside the blood into the tissue. And the opposite thing is happening into the alveoli. 4 ml carbon dioxide is unloaded from the blood into the alveoli, and 5 ml oxygen is loaded to the oxygenated blood into the alveolar level. I think you got it. So that is the importance of the partial pressure. Now let's talk about the respiratory surface. While you are talking about respiratory surface, we must need to talk about the alveoli, right? So we know very well that the alveolar epithelium is made up of thin flat cells which we are calling simple squamous epithelial cells. It is alveolar epithelium, and they are surrounded by what? They are surrounded by blood capillaries or alveolar capillaries. Now the inner lining of the alveolar capillaries, they are also made up of thin flat simple squamous epithelial cells that we call endothelial cells, and in between them, in between the alveolar epithelium and the endothelium of blood capillary, we have a non-cellular layer that we are calling basement substance, which are made up of basement membrane. Okay. So respiratory surface, so this surface we are calling it respiratory surface. The oxygen is entering through the surface and the carbon dioxide is exiting through the surface. That's why we are calling this surface as respiratory surface. So that surface is made up of two cellular layer and one acellular or non-cellular layer. So first, from alveoli, it is alveolar epithelium, then on non-cellular acellular region in between alveoli and blood capillary, that is basement substance. This basement substance basically secretion of the alveolar cells and the endothelial cells or the blood capillary cells. This is made up of ground matrix, this is made up of some fibers, and the third layer is endothelium of alveolar capillary. In the tissue, you will be finding into the alveolar epithelium simple squamous epithelium, endothelium also made up of simple squamous epithelium, flat cells, and the basement membrane or the basement substance, actually the component here that you are calling basement substance, and they are making the basement membrane. So there is a basement membrane towards the alveoli, there is a basement membrane towards the endothelium. Basically that is acellular. That was all about the respiratory surface, and now we have to talk about the thickness of the respiratory surface. So these three layers is ultra thin. Properties of respiratory surface, it should be according to the NCERT less than 1 millimeter thickness. Altogether these three layers should be less than 1 millimeter thickness, and the actual thickness is 0.2 millimeter. Okay, and we know that there are 300 million such structures are present to increase the surface area, and the surface area is near about 70 square meter, that is near to a tennis court. So that helps us in a sufficient manner to exchange oxygen and carbon dioxide inside our lungs. So our lung is quite folded. Our lung is a spongy structure which is floating into a airtight cavity, that is thoracic cavity, and our lung is a vascularized bag. That means our lung is supplied by thousands of blood capillaries. Now the next topic, that is transport of gases. Transport of gases. Okay, so blood is the medium of transport. Blood is the medium of transport of both the gases. So here we need to talk about the transportation of oxygen, we need to talk about the transportation of carbon dioxide, transportation of oxygen, transportation of carbon dioxide. See, follow this table in very careful manner. Only 3% oxygen, just because it is having a very poor solubility, will get dissolved into plasma of blood, and rest 97%, who will be carrying 97% of oxygen, that is carried with RBC, and we know that inside the RBC it is crores of hemoglobin molecule, so it is transported as oxyhemoglobin, Hb for hemoglobin, right? Now let's talk about the carbon dioxide transport. See, majority of the carbon dioxide, that is 70% of the carbon dioxide, is transported as bicarbonate. That means in the form of sodium hydrogen carbonate or sodium bicarbonate dissolved into the plasma. Okay. 20 to 25% of carbon dioxide transported with RBC, with obviously hemoglobin as carbamino hemoglobin, as carbamino Hb for hemoglobin, and the remaining 7% is present into the dissolved form into the plasma of blood.
So you have to remember that majority of carbon dioxide is transported, that is 70%, as bicarbonate into plasma. 20 to 25% transported as carbaminohemoglobin. Okay. Now let's talk about transportation of oxygen. Now prior to discuss the transportation of oxygen, we need to talk about hemoglobin. See, when you are talking about hemoglobin, that is made up of a heme group and globin chains. And that heme group has ferrous iron Fe2 plus, and that globin chains, when you talking about the globin chains, for adult hemoglobin, it has 2 alpha globins and 2 beta globins. Okay, and for fetal hemoglobin, you will be finding instead of beta, they have 2 gamma globins. Okay, so if you talk about one unit, so if this is the heme group, it has Fe2 plus inside that, and it is surrounded by globin chain. So similarly, 4 heme groups are there inside a single hemoglobin, and having 4 Fe2 plus and 4 globin chains, 2 alpha globins and 2 beta globins. Suppose this is the hemoglobin molecule, one single hemoglobin molecules. What is the central molecule we are talking about? That is Fe2 plus, Fe2 plus, ferrous. Now this Fe2 plus is having affinity towards oxygen, so it can bind with oxygen, one oxygen molecule. So one hemoglobin molecule can catch or can bind with maximum 4 oxygen molecule or 8 oxygen atom. Remember it. And few more thing about hemoglobin. In front of hemoglobin, if you give 3 gases, one is having carbon dioxide, one is having carbon monoxide, one is having oxygen, then what will be the sequence of affinity? The hemoglobin is having highest affinity towards the carbon monoxide, then it has the second highest affinity towards oxygen, and very less affinity towards the carbon dioxide. So the sequence of affinity is like that: carbon monoxide, oxygen, then carbon dioxide. The highest affinity is towards carbon monoxide, then affinity towards oxygen, then affinity towards carbon dioxide. Now we were talking about only here how hemoglobin binds with oxygen. So here we need ferrous like Fe2 plus with hemoglobin. If you oxidize the ferrous into ferric Fe3 plus, then it's having very low or negligible affinity towards oxygen. So Fe2 plus, it can bind with oxygen happily, but Fe3, it won't be binding with oxygen. And if the hemoglobin is having Fe3 plus with it, then we call it methemoglobin. So you need to remember this. hemoglobin only with Fe2 plus ion having affinity towards oxygen. Okay. And we have seen that one hemoglobin molecule can carry maximum 4 molecule of oxygen. So in similar way, okay, 1 gram of hemoglobin we have observed that can carry 1.34 milliliter of oxygen into it. And if someone is giving you 100 ml oxygenated blood, 100 ml oxygenated blood that is having a hemoglobin of 12 to 16 gram or average 15 grams of hemoglobin into it. So inside that 100 ml oxygenated blood, you will be getting near about 20 ml of oxygen. So this very simple calculation you have to understand, that why 100 ml of oxygenated blood is carrying 20 ml of oxygen. So it's a result of this particular calculation. Got it? So we can see that hemoglobin is having 4 hands to catch oxygen. If I am catching one oxygen molecule, then hemoglobin, we can say that it's 25% saturated with oxygen. If I am catching 2 oxygen, if I am hemoglobin molecule, then I am 50% saturated with oxygen. If with 3 hands of hemoglobin I am catching 3 oxygen molecule, then I am 75% saturated, and even if I am carrying 4 oxygen molecule, then I am 100% saturated. Beyond that, I cannot carry oxygen, right? So whatever we were discussing is just given in a single slide regarding the affinity of hemoglobin towards different gases. Hemoglobin, when will bind with oxygen, we will be calling it oxygenation, rather oxidation. Okay, the bond which is created in between hemoglobin and oxygen, that is a kind of coordination bond. There is not oxidation. Okay, hemoglobin with carbon dioxide, we are calling it carbaminohemoglobin. Here the carbon dioxide get attached with the amino group of one of the amino acid of the globin chain of hemoglobin, is not with the iron. And when hemoglobin is making bond with carbon monoxide, is forming a stable molecule, a stable compound, and we are calling it carboxyhemoglobin. So when carboxyhemoglobin is produced, when hemoglobin is binding with carbon monoxide, is kind of stable molecule. Okay. Now we really need to talk about the most difficult topic of the chapter, that is oxygen hemoglobin dissociation curve. Okay. So before understanding this, we need to talk about that the binding of hemoglobin and oxygen primarily the binding of hemoglobin and oxygen, a formation of oxyhemoglobin, a formation of, you can say, the formation of oxyhemoglobin or binding of hemoglobin and oxygen primarily depends upon PO2, that is partial pressure of oxygen. So there are two terms. One is binding and another is dissociation. So what is binding and what is dissociation? When hemoglobin is having enough affinity towards the oxygen, it will form oxyhemoglobin HbO2. This binding, and when the oxyhemoglobin will get split up, we call it dissociated into separately the reduced hemoglobin and oxygen, then that is called dissociation. Now we need to talk that we are talking that mainly this is the factor, partial pressure of oxygen, which is influencing this process. But there are certain other factors who can influence the oxygen and hemoglobin formation or their dissociation. So what are they? They are partial pressure of carbon dioxide, acidity. Acidity means concentration of H plus ion, means we need to talk about pH as well. Okay. Certain other factors like temperature also influence oxygen hemoglobin association or dissociation, and also another byproduct of respiration, that is 2,3 biphosphogycerate or 2,3 BPG, that is a product of respiration. So these are factors which can influence the binding or dissociation of oxygen and hemoglobin. And after studying the oxygen and hemoglobin dissociation and association inside the body, scientists have made a sigmoid or S-shaped curve where two very important things are related: partial pressure of oxygen and the saturation of hemoglobin and oxygen percentage. So how to derive that graph we are gonna study. See, we know that inside the lungs, into the alveoli, the blood capillaries or the blood vessels are getting loaded with oxygen and unloaded from carbon dioxide. Oxygen is getting in and carbon dioxide is going out. That is making the blood oxygenated blood. And for every 100 ml of blood, we are getting 20 ml oxygen into it. And we can assume that this is the 100% oxygen, 3% dissolved into the plasma and 97% with the hemoglobin. Now as soon as that oxygenated blood is approaching towards different type of tissues, it has to unload the oxygen, whatever it was carrying. Now we will take example of 3 different tissues. For one tissue where the rate of respiration is quite normal, that we call that tissue is in normal physiological condition, NPC. Another tissue where the rate of respiration is quite higher than the normal one, and there is another tissue where it is in really need of oxygen, so that is heavy exercise. If a tissue is undergoing heavy exercise, like your muscles while you are doing gym or while you are running. Okay, so right now we are just studying, we don't have to do physical work, so we are in, our tissues are prevailing in normal physiological condition. See what happens here, when your oxygenated blood is approaching towards the tissue with normal physiological condition, then generally it delivers 25% of oxygen and retains 75%. That means now here into the tissue which is having normal physiological condition, the dissociation will be 25% and the retention or the saturation of hemoglobin oxygen percentage will be 75%. Now another tissue which is having little bit more requirement oxygen, to that very particular tissue the oxygenated blood will retain 50% of oxygen and will donate 50% of oxygen. Now here dissociation is 50% and saturation is also 50%. Now the third type of tissue where the requirement of oxygen is really high, the tissues are crying hypoxia, you need to provide more and more amount of oxygen. Now to that your body will be dissociating almost 75% of oxygen and will be retaining only 25%. So it depends upon the requirement of particular tissue, that how much hemoglobin will be carrying how much oxygen and how much oxygen will be dissociated from that and will be delivered to that tissue. So depending upon this situation, scientists, they have created a chart. Now let's prepare the chart. The one side of the chart we are talking about the partial pressure of oxygen, and another side of chart will be talking about the percentage saturation of hemoglobin oxygen. So we have observed that if a tissue is having partial pressure of oxygen is having 10 only, then the hemoglobin is having only capacity to retain 10% of oxygen. If a tissue is having partial pressure of 20, the hemoglobin will retain 25% of oxygen. If a tissue is having partial pressure 28, hemoglobin will retain 50% of oxygen. If a tissue is having partial pressure 30, hemoglobin will retain 55% of oxygen. If the tissue is having partial pressure of 40, hemoglobin will retain 75% of oxygen. If the tissue is having partial pressure 60, hemoglobin will retain almost 90% of oxygen. If the tissue is having partial pressure of 80, hemoglobin will retain almost 95% of oxygen. If the tissue is having partial pressure 90, hemoglobin will retain 97% of oxygen. If the partial pressure is even 100, the hemoglobin then also will retain 97% of oxygen. Hemoglobin cannot retain beyond 97%, so that's why we are telling that 97% is the saturation level of hemoglobin inside our blood. See, one single hemoglobin can carry 4 oxygen, but altogether 100% of the hemoglobins are not workable, are not working for us. So maximum 97% hemoglobin under high partial pressure of oxygen, they can carry oxygen, and 3% will be dissolved into the plasma. Okay. Now a very important thing I am going to tell here, if a particular tissue is having 50% saturation, so what will be the dissociation percentage? Means hemoglobin is carrying 50%. So what percentage of oxygen can be donated? So the dissociation percentage will also be 50 here. If the hemoglobin oxygen saturation percentage is 25, then how much oxygen can be donated there? Or dissociation percent will be 75. So saturation percentage and dissociation percentage, they are opposite things to each other. Like how to calculate dissociation percentage? from the saturation percentage, you have to minus 100 from it. If in a particular tissue, if we are getting that saturation percentage is 75, then what will be the dissociation percentage of that particular tissue? Means if a tissue is having partial pressure 40, in that tissue the hemoglobin will retain 75% oxygen, and what percentage of oxygen it will be delivering? 100 minus 75, 25% of oxygen, 25% of oxygen will be delivered to that particular tissue. Okay. Now simply what we have to do, we have to put the values into the chart. So for 10 partial pressure, so here this axis, x axis is for partial pressure and the y axis is for hemoglobin, oxygen, saturation percentage. So for 10 the saturation will be 10, for 20 the saturation will be 25, for partial pressure 20 the saturation will be 25. You know how to prepare a graph. For 28 it's 50, for 30 it's 55, for 40 it's 75, for 60 it's 90, for 80 it's 95, and for 90 it's 97. And that is the saturation percentage, high saturation percentage. So if you connect all these dots, whatever you are getting a S-shaped sigmoid curve. And what is the importance of sigmoid curve? First, here, if the partial pressure is low, the saturation percentage will also be low. So that's why in the beginning is forming a slope. Then with increasing partial pressure, suddenly the graph is forming a steep surface. So it's a slope, then it's a steep surface, then it will be gonna slope again. So after crossing, when the partial pressure is crossing the partial pressure 20, from 20 to 60 you can see that is the steep position, of the curve. From 0 to 20, this is the slope position of the curve. So there is slope, steep and slope, and that is the nature of S or sigmoid curve. So that means with low partial pressure, if the partial pressure is low, hemoglobin won't be showing affinity or the strong bonding towards oxygen. Keep increasing the partial pressure, now hemoglobins are efficiently catching the oxygen. So the curve is becoming steep. Now after crossing 60, here you can find that 90% hemoglobins are saturated. So again the hemoglobin will decrease their affinity towards the oxygen. So that's why after 60, here again you are finding a slope here, and when it's touching 97 is just linear graph. So that was all about oxygen and hemoglobin dissociation curve. So what it's explaining us, it's explaining that if the partial pressure is low inside the tissue, less amount of oxygen will be carried by hemoglobin. If the partial pressure is high, more amount of oxygen will be carried by hemoglobin. Okay, up to a certain limit. So that is the relationship between partial pressure of oxygen and hemoglobin oxygen saturation. Okay. Now, just I will be proving one thing here. We were telling in normal physiological condition, the partial pressure of tissue is 40. The partial pressure of oxygen in tissue is 40. We know that very well. So in 40, what is the saturation percentage? Saturation percentage we got 75%. So in normal physiological condition, a tissue is having partial pressure of oxygen 40. So in that particular tissue, how much hemoglobin will be carrying oxygen? 75% oxygen is carried by hemoglobin. So what about the remaining 25%? 100 minus 75. So the remaining 25% hemoglobin is dissociated and delivered to the tissue. We know that 100 ml oxygenated blood is carrying 20 ml of oxygen. Now under normal physiological condition, it delivers 100 ml oxygenated blood. If it's carrying 20 ml of oxygen, under normal physiological condition, it will be delivering 5 ml of O2 to the tissue. Hence it's proved from this chart. Now if you find a tissue where the partial pressure of oxygen is 20, that means heavily exercising tissue, then what will happen into that tissue? So you have to calculate that in that tissue the hemoglobin can carry only 25% of oxygen. So in heavily exercising tissue, hemoglobin can retain 25% oxygen. So 75%, 100 minus 25, 75% of oxygen will be delivered or released out. So under heavily exercising condition, that 100 ml of oxygenated blood which is carrying 20 ml of oxygen will not be delivering 5 ml of oxygen to the tissue, will be delivering 15 ml of oxygen to the tissue and will be retaining 5 ml. So that's how this curve will be helping us, that how our oxygenated blood is deciding that which tissue to give what amount of oxygen upon its necessity. Got it? Okay. Now let's talk about some left shift and right shift. So if this is the standard curve, sometime we can observe the curve is shifting towards the left, sometime you observe that the curve is shifted towards the right. So what is the importance of right shift and what is the importance of left shift? Let's discuss. So we were discussing there are certain factors which like create an environment inside our body, that when and why hemoglobin will bind with oxygen to form oxyhemoglobin, right? So what are those factors? Those factors are partial pressure of oxygen, partial pressure of carbon dioxide, H plus ion concentration, acidity, pH, temperature, and presence of 2,3 bisphosphoglyceric acid. See, now listen it very carefully. If there is high partial pressure of oxygen, low partial pressure of carbon dioxide, low concentration of hydrogen, low acidity or less acidity, high pH, acidity low means pH is alkaline, low temperature and less respiration means less formation of BPG. Under these circumstances, the curve will show left shift. Why? The left shift, that means good condition of your body. That means high oxygen, less carbon dioxide. And exactly the opposite, if you create the opposite scenario, low oxygen partial pressure, high carbon dioxide partial pressure, high concentration of H plus, high acidity, low pH, high temperature, high BPG, the curve will shift towards the right condition. The right shift means your body is vulnerable. Maybe some accident took place, maybe you are suffocated, maybe you are in a bad condition. So left shift will suggest good condition of our body, right shift suggests bad condition of your body. Now how to calculate that? By calculating the P50 value. What is P50 value? The partial pressure of oxygen where the partial pressure of oxygen where hemoglobin oxygen saturation percentage is 50%, that is called P50 value. Okay. So suppose for standard curve we got the P50 value was 28 partial pressure. If your tissue is having partial pressure of 28, then 50% of hemoglobin will be carrying oxygen. Okay. Now if you observe a left shift, then the P50 value suppose decreases from 28 to 15. So if the P50 value decreases, that means that will create a left shift. So that will decrease. P50 value denotes increased affinity of hemoglobin towards oxygen. That means left shift, that is showing increased affinity of hemoglobin and oxygen. Now if you calculate the P50 value for the right shift curve, suppose the P50 value has increased from 28 to 48 here. So that means if you have to increase the partial pressure of oxygen inside your body to accommodate 50% of oxygen into your hemoglobin. So that means decreased affinity. Affinity means how eagerly, how strongly hemoglobin is wanting to form coordination bond with oxygen. So left shift increased affinity, right shift decreased affinity, and these are the condition which can promote the left shift of the curve or the right shift of the curve. Okay. Now one question is given here. There are 4 curves labeled as A, B, C, D. Now you have to find out which curve is for male, which curve is seen in female, which curve is seen in fetus, and which curve is for our myoglobin. See, if you compare male and female, who is having more blood? Yes, due to the formation of testosterone hormone, males can produce more blood and RBC and hemoglobin. So more hemoglobin is more affinity towards the oxygen. So this curve C will be for male. C and D, suppose I am comparing between which one is male, which one is female. So here curve C, that is towards the left, that is for male, and curve D, that is for female. Why? Because female has less amount of hemoglobin. Now we know that fetal hemoglobin is having higher affinity. in compared to adult hemoglobin. So curve B is explaining that this curve is for fetal hemoglobin, as because in fetal hemoglobin it has 2 alpha chain and 2 gamma chain instead of beta chain, and that gamma is having more affinity in compared to beta chain towards the oxygen, right? And myoglobin, the curve A depicting the curve or hyperbolic curve of myoglobin, that is having a pigment which is present inside our muscle that we call myoglobin. Like hemoglobin, they are not 4 unit, they are single unit molecules. They are having highest affinity inside our body. Okay, so inside our muscle we have myoglobin, and they will be forming a hyperbolic curve when we talk about the oxygen affinity. Now let's talk about the transportation of carbon dioxide. We know how carbon dioxide is transported, the basic table we are knowing. So what happens here, from tissue, suppose these are tissue cells, continuously the carbon dioxide will be entering into the RBC. And here, for kind of information, the RBC is carrying a very important enzyme called carboxy anhydrase. Though anhydrase enzyme is present also in plasma. So this is RBC and this is plasma. So while this carbon dioxide will enter, so first of all, the what you can observe, that 7% of carbon dioxide is already dissolved into the plasma. So now let's talk about the transportation of carbon dioxide. So when blood is becoming deoxygenated, remember, when the carbon dioxide is entering into the blood, it will make the blood what? Deoxygenated. Now how that blood is produced and how carbon dioxide is transported with it, along with it. So 7% of carbon dioxide directly get dissolved into the plasma. Okay. The remaining carbon dioxide that will enter into the RBC, and what I was talking, that I was telling that that RBC is having a particular enzyme called carbonic anhydrase. Though this carbonic anhydrase is present into plasma as well, but more concentration you will be finding inside the RBC. Now, as soon as the carbon dioxide enters into the RBC, it will react with water by the help of this biocatalyst carbonic anhydrase and will get carbonic acid, H2CO3. Now, as soon as carbonic acid is formed, again that monkey carbonic anhydrase will come and it will break down our carbonic acid into H plus and HCO3 minus. Now here we got a proton and a bicarbonate ion. Okay. Now what is the fate of this bicarbonate ion? there are certain transporter protein present on the membrane of RBC. We call it transporter band protein 2. So this transporter protein will be throwing out this bicarbonate from RBC to plasma, and as soon as the bicarbonate they are entering into the plasma, we know that the plasma is having Na plus and Cl minus in the form of salt, right? So that Na plus will come and will bind with bicarbonate ion and it will form sodium hydrogen carbonate. So inside the sodium hydrogen carbonate 70% of CO2 you will be finding. That is transported in the form of 70% CO2 inside this compound sodium hydrogen carbonate. So NCERT is saying maximum or majority of carbon dioxide is transported in the form of bicarbonate ion. Now after some time what the RBC will feel? There is more H plus ion and it's kicking out all the HCO3 minus. Now it need some negative ions. Now it needs some negative ions or anions to replace HCO3 minus. So chloride ion Cl minus, it was crying alone inside the plasma. So that chloride Cl minus will be transported as replacement to HCO3 minus inside the RBC, and that will react with potassium ion and will form KCl. That's how the ionic balance is maintained, and this particular step we are calling it chloride shift. And where you can observe this? Into the tissue level blood. Okay. Now what about the remaining 23% carbon dioxide? See, if there is more proton produced inside the RBC, it is a threat for RBC. So now hemoglobin will come. That hemoglobin will catch this monkey kind of ions called protons and will form hemoglobinic acid HHB. Now these hemoglobins are acidic in nature. Now as soon as that hemoglobins are acidic in nature, they will lose the affinity towards the oxygen and the oxygen will be thrown out from the hemoglobin. Now what was the source of production of this H plus ion? That is due to the entry of carbon dioxide, and this is, we are calling it Bohr's effect. The dissociation of oxygen from hemoglobin in tissue due to the high partial pressure of carbon dioxide and presence of H plus ion. So as soon as the HHB are formed, this acidic hemoglobin will be releasing oxygen, and this oxygen from RBC will get dissociated from hemoglobin, being dissociated to hemoglobin will enter into the tissue to perform the respiration, cellular respiration. And now this acidic hemoglobin is having a good affinity or high affinity towards carbon dioxide. So it will bind or it will take the carbon dioxide with the amino group with one of the amino acid of the globin chain of hemoglobin, and it will form carbamino hemoglobin. So here you can see 23% of carbon dioxide. So that's how carbon dioxide is transported inside your deoxygenated blood, and whatever we were discussing, that is given, everything is written here in this particular slide, right? So again we will be discussing one more important point. What is Bohr's effect? though it's related to oxygen and hemoglobin dissociation, but without discussing that carbon dioxide transport, it will not be more or much relevant to us to understand. So Bohr's effect, the scientist Bohr, there is different scientists from Niels Bohr of chemistry. This particular scientist Bohr is saying that dissociation of oxygen and hemoglobin takes place into tissue blood due to high partial pressure of carbon dioxide and high concentration of H plus ion, and also along with that obviously in tissue we will be getting low partial pressure of oxygen. So that is Bohr's effect. So it's just opposite phenomena what's happening into the alveolar level. So in alveolar level you will find a different effect. This is called Halden's effect. Here the Bohr effect, he is telling about the tissue level. The Halden, the another scientist, he will be telling, he will be describing whatever the things or chemical reactions happening into the alveolar level, right. So our relevant the relevant discussion is Bohr's effect, though directly NCERT is not telling about Bohr's effect, but we need to learn for sake of our exam purpose. Okay. So that's what we were studying, the major fraction of carbon dioxide given out readily react with cytosolic H2O in RBC due to the presence of carbonic anhydrase. Again you have to remember the name, the carbonic anhydrase enzyme which is present inside the cytosol of RBC. Okay. Now what is the difference between CO2 carbon dioxide transportation in tissue level and carbon dioxide transportation in alveolar level? In tissue level we can observe chloride shift. In alveolar level we can observe reverse chloride shift. In tissue level, why the carbon dioxide is getting loaded and oxygen is unloaded? Due to the high partial pressure of carbon dioxide, low partial pressure of oxygen and high concentration of H plus ion. In alveolar level, the opposite phenomena, the carbon dioxide is getting unloaded and oxygen is getting loaded into the blood. Why so? In alveolar level, there is high partial pressure of oxygen and low partial pressure of carbon dioxide, just opposite, and there is very less concentration of H plus ion. So that's why we were telling that in tissue level we can observe Bohr's effect. In alveolar level, when there will be association or the binding between oxygen and hemoglobin due to high partial pressure of oxygen and low partial pressure of carbon dioxide, that we call Halden's effect. And here you can find the reaction. When you are talking about tissue level, the carbon dioxide will associate with hydrogen and will react with the help of carbonic anhydrase enzyme to form H2CO3. Sorry for this. carbonic acid, and again carbonic anhydrase will break down or split the carbonic acid into HCO3 minus and H plus. Okay, and if you study the backward reaction, that's happening where? That is happening into the alveoli. Inside the alveoli, what will happen? H plus and HCO3 will bind together to form HCO3 minus again. Second, again this carbonic anhydrase will break down HCO3 into carbon dioxide and water. The carbon dioxide, the water will remain inside the RBC and the carbon dioxide will get diffused out from the deoxygenated blood and into the alveoli. So the forward reaction, so this reaction is reversed. The forward reaction that is happening into the tissue and the backward reaction that is happening into the alveoli. Now the second last topic of the chapter, that is regulation of respiration. See, listen it very carefully, my lovely future doctors. So when you are talking about regulation of respiration, there are mainly two types of regulation is seen inside our body. One is the neural regulation and another is chemical regulation or chemical regulation. Now when you are talking about neural regulation, some parts of brains are obviously responsible for neural regulation, and the parts or the nucleus or the center, they are present inside the major center, they are present inside the medulla oblongata. And we call that center respiratory rhythm center. It is the master regulator of normal respiration, and we have some neural centers present into our pons. We call them pontine centers or pontine nucleus. They directly cannot regulate the respiration, but they will modulate or moderate the rate of respiration. They can either increase or decrease the rate of respiration by giving signal to this respiratory rhythm center. So pons, when you are talking about pons, it has one pneumotaxic center and one apneustic center. Again I am telling which is the master regulator. Respiratory rhythm center is the master regulator for our neural respiration, neural regulation of respiration. When we are talking about pons, it's not directly regulating the respiration or breathing rate. With the help of pontine center like pneumotaxic center or apneustic center, either they can increase or decrease the rate of breathing by giving signal to the respiratory rhythm center. So they are regulating or moderating via RRC, respiratory rhythm center. Okay. Now what about chemical regulation? See, this is a very conceptual thing. Carbon dioxide and H plus ions, they are responsible for chemical regulation because we have chemosensitive areas inside our body and they can sense carbon dioxide either or H plus ion. Role of oxygen is insignificant in regulation of respiration. Role of oxygen is insignificant in regulation of respiration. Remind you, role of oxygen is insignificant. In regulation of respiration. Okay. Now, where you will be finding those chemosensitive areas? One chemosensitive area you will be finding near to respiratory rhythm center. There is one chemosensitive area, and there are other chemosensors. So, this chemosensor, they are present there. This is the part of central nervous system, and there are some other chemosensors which are part of your peripheral nervous system. Okay, what are they? Aortic and carotid bodies. They are part of your peripheral nervous system. Okay. So these chemosensors, they can sense carbon dioxide concentration and H plus ion concentration inside our body. They cannot sense oxygen. I am repeating it. These chemosensitive areas, they cannot sense oxygen. That's why the role of oxygen is quite insignificant in regulation of respiration. And where these chemosensitive areas are present? One chemosensitive area present near to respiratory rhythm center of your medulla oblongata, and few other chemosensors or chemosensitive areas are present inside the aortic arch and carotid arteries. We call them aortic bodies inside the aortic arch and carotid bodies which are present into the carotid artery. Okay. Now we'll be disclosing about all the things or the important concepts about this diagram. So here we can see that this is our brain, obviously, and this part is medulla oblongata, right? And in medulla oblongata you will be finding special zone or nucleus that we are calling it respiratory rhythm center, RRC. And there are two important zones present in respiratory rhythm center: dorsal respiratory groups and ventral respiratory groups, DRG and VRG. We won't be going into that depth because it's not given in NCERT. Just remember, the master regulator of respiration or neural regulation, master regulator of neural respiration center is called respiratory rhythm center. Where it's present? It's present into the medulla oblongata. And there are other neural regulatory centers which directly cannot regulate our respiration, but obviously can give signal to the RRC to moderate the rate of respiration. You will be finding them into pons. This is also part of your hindbrain. So in pons you got one center that is called pneumotaxic center, and you got another center that is called apneustic center. Okay. Now what is the function of the pneumotaxic center? Function of pneumotaxic center, it will be reducing the duration of respiration. Pneumotaxic center, there is called switch off point for inhalation. Switch off point of inhalation. So whenever we are taking deep breathing, deep inhalation, deep inspiration, so it will cut it down. So it helps us, pneumotaxic center, to increase the rate of breathing, and it decreases the depth of breathing. Pneumotaxic center, it increases rate of breathing and decreases depth of breathing. What is the function of apneustic center? Opposite to the pneumotaxic center. Apneustic center, it will be decreasing the rate of breathing. Apneustic center, it will be decreasing the rate of breathing. Okay. So that was all, and also recapitulation of chemosensitive areas. they are sensitive to carbon dioxide and H plus ion. One center near to respiratory rhythm center, one center into the aortic bodies. Now I am explaining it. What is aortic bodies and carotid bodies? So this is aortic arch, right? And from left ventricle, blood, the oxygenated blood is pumped throughout the body. Now before pumping, if you have the measuring chemosensitive areas inside the aortic arch, the aortic bodies, it will be really easy for our neural regulatory center to measure it in a proper manner. Okay. And here you will be finding some carotid arteries which are taking that oxygenated blood towards your brain. So inside that arteries also you will be finding some chemosensitive areas. So obviously our brain is the highest regulatory center of our body. Our brain has that respiratory regulatory center RRC, right? So before reaching the blood is before the blood is reaching towards your RRC, a chemical input is taken inside your carotid arteries, which are responsible for sending the oxygenated blood to your brains. Okay. Now we'll be concluding the topic with 3 disorders. Okay. The first disorder is asthma. Now what is asthma? What are the key points of asthma? See, first key point is difficulty in what? Difficulty in breathing. Second key point, wheezing sound. Second key point, wheezing sound. And third key point, that is inflammation. Third key point, that is inflammation. Okay. Now why it happens so? Basically this is difficulty in breathing. While we are inhaling or exhaling, our respiratory system will produce a sound like a broken speaker, that is called wheezing sound. So this kind of sound is called wheezing sound. You might have observed asthma patient. Now why it's happening? See, this side you can see, this is the bronchiole of healthy person or healthy lung. But for a asthma patient, due to the inflammation inside the bronchi and bronchioles, the lumen of bronchi is swollen up, so the surface area of lumen has decreased. So ultimately the passage of air passage inside your bronchi and bronchioles is choked. That's why whenever you are inhaling or exhaling, that is creating problem for you and producing wheezing sound. Now what is inflammation? Pain, swollen up parts, swallowing, redness, pain, and deposition of fluids. So all these things together we will be calling it inflammation. And for inflammation, who is responsible here? Maybe some allergens, okay, maybe your autoimmunity, your own immune system, and obviously excess production of histamine. Excess production of histamine is also responsible for inflammation. Okay. So histamine, basically that is a vasodilator but bronchoconstrictor. That is the root cause of inflammation. So if you want to get rid from the allergy, what you need to take? You need to take some inhalation or some inhalation of some drugs or chemicals or medication which are antihistamine in nature, right? Or which can suppress your immune activities there. The second disorder is emphysema, that is a part of COPD, chronic obstructive pulmonary disorder. Okay. Now there can be many reasons for this emphysema or COPD, but one of the major reason is, yes, foo-foo, the cigarette smoking. Now while you are smoking cigarette from long term, so that will gradually create obstacle, that will gradually kill your pneumocyte cells inside your alveoli. So that's why it's telling it's a chronic disease. Chronic means which is like growing inside your body from a long time. So long term diseases are called chronic disease. Now see the difference between healthy alveoli and affected alveoli. So cigarette smoking not only causes cancer, it has many aerosol particles. Okay. So what they are doing, when they are getting inside your respiratory passage in the form of cold air, that will start disturbing your pneumocyte 1 and pneumocyte 2. So ultimately it will cause alveolar damage of your alveolar wall. So when alveolar walls and alveolar cells are damaged, that will cause decreased respiratory surface, and if the respiratory surface is decreased, that will be really like difficult for your body to supply enough amount of oxygen to you, and again your body will be facing or suffering from hypoxia and all. So ultimately it may cause choking of your lungs, shrinking of your lungs, and ultimately it may cause death as well. Okay. So the disease, remember the disease emphysema. Okay. Now the last disease, that is ORD, occupational respiratory disorder. Now these are the disorders. What is disorder basically? A group of symptoms you can observe here. There is no particular disease we are talking about, a group of symptoms, a group of diseases, and some unknown factors are responsible for that. Okay, obviously there is a term syndrome is also there when you will be finding group of symptoms, but disorder, that means obviously group of symptoms are there. You are you cannot identify what is the disease actually, what is the root cause. Sometime you may guess it, sometime you may not guess it. So there is a question mark before the symptoms or different type of multiple types of symptoms. So that kind of problems we are calling it disorders. So occupational respiratory disorder means these are the problems coming from your occupational, which are related to your job or workplace. So specifically the persons who are working in grinding or stone breaking industries. So what will be happening here, due to the excess dust inhalation, the dust particles will create or will promote formation of fibers into your basement membrane, that is called fibrosis, and ultimately that will create a long term inflammation in your lungs, and the diseases may be caused by silicon particles, and we will be calling it silicosis. Asbestos particle, we may call it asbestos. Even those who are working in collieries affected by coal particles, you may get black lung diseases. So that was all about today's lecture and the chapter breathing and exchange of gases. So in coming time I'll be coming with more chapter with more detailed explanation. So for today it's a thank you for everyone. Bye bye and Jai Hind.